Prasanna, your name came first.
Babu, you're planning to come to Estere. Are you coming to Estere?
Oh, I was planning to come to your country as well, but till
Yes.
date I haven't got my visa. I had applied last month on 22nd. Until now, I don't have. So people are making fun that God doesn't want you to go to Europe this time.
To Greece? Oh.
Yeah, I haven't got my visa yet.
Okay.
Maybe you'll get it.
But we have time yet, so you may have it within the next week, maybe.
But he has promised me that they'll deliver it before 6th of June because I had to apply for UK as well. But till date, I haven't got it. It's never happened before. This I used to get it within a week. I don't know what's happening.
Why do they say there is so much delay?
They
Why?
are not telling anything.
That's a pity. You cannot plan without with them being so.
Yeah, 27th I have to leave, and till date I haven't got it.
I think you'll get it because you have given the registration letter, no, Ishri?
No,
They
I
give-
did apply through Helsinki.
Ah, okay. Will it roll those
If you are
ideas?
coming, please let me know so we
I'll
make.
Dr. Srinivas has joined.
We are 402.
Srinivas sir join ಮಾಡಿದ್ದೀರಾ ನಾನು start ಮಾಡ್ತೀನಿ.
Yeah, I joined.
Okay, we
thanks.
are starting.
Yeah.
Seema. Dr. Seema, we are starting.
Hi, boss.
Hi, hello.
I will
Bye,
start.
bye.
Hello.
Bye.
I was telling Usha hi.
Hello, madam.
Hello.
Seema,
Hi.
you start the welcome and then I'll take over.
So,
Okay.
Kiran is not going to speak before me?
No, where is he? He's there? Corona. Mehul is there, I think. Mehul,
ma'am.
you are there, no?
Yeah.
Mehul, will you speak or I start?
No issue. You can start, ma'am, directly. Otherwise, I will introduce you. Then we can start.
Ah, that's better. Chalo.
Yeah.
Let's
Okay.
do
No issue. So, welcome all the faculties and delegates for the Core Connect live webinar, which is on the Insight series and Evening with Hisarians. So, first of all, it's my honor to introduce our conveners, Dr. Seema Pandey and Dr. Anuradha Khar. So, Dr. Seema Pandey is the librarian of ISHAR, and she having the National Executive Committee member of ICOG and ISPAT. And Madam is also co-author of A to Z PCOS, everything you wanted to know about the PCOS, and co-editor of the ART and fertility enhancing surgeries, the Synergy Ovulation Induction Simplified, and Foxy Handbook of Drugs in Infertility and Foxy Focus Preterm Labor. Madam is the reviewer of the Journal of Obs and Gynae India, and also having the key interest in infertility and reproductive endocrinology, PCOS, adolescence, and early pregnancy failure. And Madam is also spreading awareness about PCOS and adolescents health among the rural women and school girls through health checkup camps and talks through her NGO and Survey. So welcome, ma'am, and hand over this webinar, this session to you.
Thank you, Mehul, for a kind introduction. Good evening. Can you just stop sharing the? Can you stop sharing the screen? Yeah.
Yes, ma'am.
At the outset, I would like to welcome you all in this Insight series of An Evening with Isarian. This is our second episode where we are focusing totally on genetic part of especially male infertility. So I would extend my warm welcome to our esteemed faculty, Dr. Charudat Joshi, and guest of honor and chief guest, Dr. Charudat Joshi, Dr. Srinivas, our esteemed faculty international speaker, Dr. Alexia, a very good friend. Thank you so much. Dr. Rahul, Dr. Manisha, and all the other faculty. And dear viewers, we are trying to bring up a topic which is really, really important for our day-to-day clinical practice. At the same time, where a lot of dilemmas and Dr. Usha Dave, welcome to Dr. Usha as well. So the kind of faculty we are having, these are here to just break certain myths and give you proper take-home messages regarding what's new, because future is genetics, stem cell, and all those innovation, and not to forget AI. So with this, I would welcome you all again to this insight and hand over this platform to my dear friend and co-convener, Dr. Anuradha Khan, to carry it forward.
Thank you, Dr. Seema. First, I would like to read the CVs of our chief
Opening
guests.
prayer.
Yeah, I thought I'll welcome them and then we do the prayer.
Okay, okay, please do.
Yeah,
Go
let
ahead.
them.
Yeah.
Yeah. Can you please share their CVs? Yeah. So we all know Dr. Charit Joshi, who is the chairperson embryology at present, ISAR. He has done post-graduation in life sciences, doctorate in life sciences. He's a trained ART and allied procedures at Belgium, Singapore, Germany. He has served as a lot of office bearer, as an office bearer for a lot of societies, and he is the past vice president of ACE and medical director, Genes India ART Bank, Indore. He is the lab director of RD Gadi Medical College, Ujjain. And at present, he is our chairperson embryology. Sir, welcome, sir. Our guest of honor is none other than our Dr. M.S. Srinivas, who has been my teacher too. Welcome, sir. He is director and principal embryologist, Carey Fertility Private Limited. Is the founder, president of ACE India, course director, MSc in ART, Mysore University. He has been the past president of embryology ISAR and scientific director of Narayana Health. And he has also been our past president at KISAR, and we've learned a lot from him. He's an ICMR expert committee, ART guideline and ART bank. Welcome, sir. I would like now to do a small prayer before we begin this academic program.
असतो मा सद्गमय तमसो मा ज्योतिर्गमय मृत्युर्मामृतं गमय ॐ शान्ति शान्ति शान्ति।
So,
या कुन्देन्दुतुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा।
I would like first to give our condolences to our citizens and citizens from other country who lost their lives in the Air India crash recently, and may their souls rest in peace and Om Shanti. And now we start our discussion and we move on to our first talk of the day, unraveling the genetic basis of non-obstructive azoospermia. And our speaker,
Anu, can I just interrupt? Before you start the talk, we would like to hear our chief guest and if he would like to say a few words. Dr. Charu.
Thank you, madam. This is a wonderful event you are organizing, and this is need of the day today. And I think after Corona, this is the best platform to interact and understand each other and to learn from each other. So, without taking much time, I would just like to say this is the topic which is chosen today. It's a recent one, and lots of happenings are there in the world about this genetics and PGD and all, and maybe in future AI integrated with genetics and so many things we are expecting in our field of ART. So very eager to listen the expert views and basics and developments what are happening in this field. So thank you once again for inviting me as a chief guest here, and all the best. Thank you.
Thank you, Charu.
thank you, sir. Dr. Srinivas?
Yes.
Yes, sir.
Good
Of
afternoon.
course.
Thank you so much for inviting me as a guest honor. And you people are, Seema Pandey, both of you and Dr. Anuradha, taking care of the historian by conducting this the webinar, which is going to be really useful. You know, we are very least bothered about male infertility. There is no empirical treatment for the male infertility. So this is one occasion so where many of us will get to know the more on how we'll be able to deal with the male infertility because as an embryologist, we all have to get one sperm, whether it is epididymis or the testis, by you can cook, do it. But there are certain things which we did not understand, the genetic part of it. So it is good that you people are bringing it out. I wish you good luck.
Thank you, sir. I would like to invite for our next talk, which is unraveling the genetic basis of non-obstructive azoospermia. I would like to first invite the experts, Dr. Usha Dave and Dr. Pratik Makwana. Dr. Usha Dave, she has established the Center for Research in Mental Retardation in 85, started her first GCMS metabolic testing services in India in 97. She has received ISPAT Lifetime Achievement Award in 23, National Award in the field of genetics in 24. She's the national president. She's been the national president ISPAT 21-23 and VP MPAI in 2018-25. She's the founder member of ISEM, Indian Society of IEM, and State Appropriate Authority and Advisory member of the Government PCB&DD Act in Maharashtra State, board member of Japanese Society for Biomedical Mass Spectrometry. And she has more than 150 publications in national and international journals and books, and has done a lot of research projects with ICMR, UNICEF, AMDA, MAILS, Birla Smart Kosh. And I think if we start telling about her, we'll take like 10 minutes to just describe her. And her specialty is clinical biochemical genetics, cytogenetics, molecular DNA diagnosis, and specialty mass spectrometry applications, neurodevelopmental and reproductive genetics, and community genetic healthcare and genetic counseling. And she has one book to her name, Genetic Counseling, Clinical and Laboratory Approach, as an editor. So welcome, Dr. Usha. I would like to now introduce our second expert and hand over to our experts. Dr. Pratik Makwana. He is Director of Vasundhara IVF and consultant embryologist, healthcare educator, and content creator. He is the member of WHO FIDES, Eshrey Young Ambassador 22, a TEDx speaker, former Advisory Board Council on Sexual Reproductive Health and Gender Education. He's authored several chapters in textbooks of ART and IVF, multiple papers, poster presentations in national and international conferences. He's been faculty at a lot of national and international conferences. He has top 20 Healthcare Communicating Award, ISW Council, Niti Aayog, and National Health Authority. Best Debater in 40 Vision 21, Best Paper under 40 19, Reproductive and Sexual Health Educator and Influencer. And he has a huge fan following, huge subscribers, and he's a big influencer. And ISAR Achievers Award 23 and IFS Achievers Award 23. Welcome you all. I would now hand over to Dr. Usha, and could you please introduce our speaker, Dr. Alexia?
Thank you, Dr. Anuradha. Welcome, Dr. Alexia. I'm very keen on looking forward to your talk. I came to know about you through Dr. Seema and Anuradha. Here we have the person who is already working in the risk management professional diploma she has. She is embryologist, European Society of Human Reproduction, embryo. Professional affiliation, you can see over here, she has European Society, American Society of Reproductive Medicine. She is a scientist in reproductive. Her career highlights are, we can see over here as the presented TED Talk to the 10 TEDx University, Texas, is it? Helsinki. 20 obtained PMI Risk Management of Professional Diploma 2019. She has a PhD thesis. Oh, that's the genetic basis of azoospermia. So we look forward to unravel, and the list will go on. She's a lab director, of course, of Embryo Lab. And we are all very keen over here from the embryologist and who has hands-on skills, with tremendous experience. We look forward to hearing from you. Alexia, the dais is yours.
Thank you so much for this amazing introduction, and it's a great honor to be among you all with all these quite long and lengthy achievements, and many congratulations to all of you. So I would like to thank the organizing committee and my dear friends, Dr. Anuradha and Dr. Pandey, Seema Pandey, for having me, and all of you, of course, for having me among you and be part of this evening, the ISARIAN's evening. So as Dr. Dave said, I'm going to elaborate and discuss on the genetic basis of non-obstructive azoospermia. And as you pointed out, this is my completion of my PhD, and I'm going to present you a part of our findings. Because being actively involved in embryology with a deep interest in azoospermia, it was for over 30 years, it was a huge challenge for me to provide the best we can to these men diagnosed with non-obstructive azoospermia. And as you pretty much know, the answers to this, the diagnostic or treatment answers to these people are very limited. The only option is biopsy. And even if we do biopsy, we never know whether this is going to be a successful or non-successful outcome. So pretty much the challenges were there. So I started investigating the genetic aspects of this pathology, and I'm going to present to you my experience, because obviously when you take a look from the genetic point of view, things become more clear. So I have no conflict of interest, just a scientific interest, and as you already know, azoospermia is the total lack of spermatozoa in the ejaculate after in at least 2 sperm samples and after centrifugation. The prevalence of this quite challenging condition for male infertility is estimated to be 1 out of 10 infertile men. More or less, it represents 1% of the total population globally. And this means that millions of men suffer or are diagnosed with azoospermia. And currently, we know that there are the causative factors may lay on a pre-testicular level, like a hormonal disorder, or testicular level, a disorder on at the level of spermatogenesis, or in a post-testicular cause, which may be due to an obstruction on the roots, a mechanical issue after the testicular environment, the well-known obstructive azoospermia. So unfortunately for us and for the patient, the most common form of azoospermia is what we call non-obstructive azoospermia, where there is not necessarily any hormonal dysregulation involved or an obstruction, but there is a severe impairment or a total lack of spermatogenesis. And this represents the 70% or more of all azoospermia cases. And unfortunately, currently, with the current state of knowledge, only in 25% of these men a sound genetic diagnosis is available, while in the rest of the 75%, there's no genetic, non-genetic issue or a secondary finding that can explain the lack of spermatozoa in the semen. So this kind of azoospermia, which is considered the most challenging type of azoospermia, is termed idiopathic. So there is no clear diagnosis for these men. And apparently it seems that in most of the cases, non-obstructive azoospermia is just a symptom of a wide variety of different factors, most of them of genetic origin. Until 1992, which is my starting year as a clinical embryologist, azoospermia was considered synonymous to sterility. It was considered untreatable. And it was only thanks to the milestone introduction of ICSI, intracytoplasmic sperm injection, into the clinical practice back in 1992, followed by the first use of testicular spermatozoa back in 1993, and the first baby born after the use of testicular spermatozoa in 1995, where the situation changed forever. And for the first time in the history of the humankind, these men could have a hope of fathering their own biological child. And since then, and we are counting already 30 years of practice, these men follow the route for non-obstructive, the roadmap of the non-obstructive azoospermia, which involves the investigation, sperm tests, scanning tests, genetic tests, before they move on to a kind of pre-treatment if it is considered necessary, such as medication, vitamins, or intervention like varicocele repair. But in most of these cases, in the vast majority of these cases, the next step is biopsy, because this is the only way to see and explore whether there is an active spermatogenesis in at least some of the locations of the testicles. So in order to maximize the sperm retrieval for these men, there has been a variety of different intervention approaches, which may involve the FNA or the testicular biopsy or the microtest. And once we have a positive outcome, these men can use the sperm in the fresh or frozen state to proceed to ICSI in a hope that there will be a successful outcome, or in case of a negative biopsy outcome, then alternative options may be explored. over the years, and while we got to become more and more experienced in treating in non-obstructive azoospermia, we came to realize that microTESA seems to represent the best possible way to investigate the testicles for these men, as we have a kind of mosaic state in the testicular environment, and spermatogenesis may take place in only some loci in the testicle. So it's a much better option to investigate thoroughly and map all the testicles before we are sure that there are no or there are spermatozoa. And add to that, we have an increased efficiency in the processing of the testicular tissue samples. So the better we become, the more we increase the recovery, the sperm recovery rate. So once we have the testicular spermatozoa, as we said, we are able to use them in fresh or frozen state. And in the best possible scenario, in the best case scenario, it seems that out of these men that undergo biopsy, we have approximately 50% positive recovery rate, which is good news. If we go back in 1992, where none of them had any option. But imagine that in 50% of the cases, men undergo an unnecessary intervention because we cannot make sure before that whether there is or there is no active spermatogenesis.
All the delegates, kindly mute yourself.
Sorry?
Can you see my slide?
Yes.
Yes, ma'am.
I'm sorry.
Sujata?
Yeah, please mute everyone else except the speaker, Mr. Mehul.
So we have 30 years of ongoing clinical practice for non-obstructive azoospermia. And while we are happy that in at least half of them, we are able to provide an option, a treatment option, in a hope that they will have a successful outcome in terms of achieving biological babies, their own biological children, we are pretty much aware that now this approach is not anymore enough, because we have to deal and meet our challenges. There are plenty of challenges when treating non-obstructive azoospermia at a diagnostic level. Remember, 70% of them, 75% of them are termed idiopathic. At a therapeutic level, the treatment options are limited. It's rare that we have a fully recovery of spermatogenesis with any of the available treatments so far. The treatment option, biopsy, remains the main option to proceed to investigation whether there is or not active spermatogenesis. The exit challenges, even if we do recover spermatozoa, we cannot assure the couple or the man that these spermatozoa can achieve a viable and a healthy embryo, because there are lots of pieces of information and knowledge that are still lacking. And last and above all, what do these people, these men, with the use of their spermatozoa convey to the next generation? If there is indeed genetic basis involved in non-obstructive azoospermia, what is the impact on the next generation? Over the years, there is an ongoing research on the genetic field, and we pretty much know today that there are thousands of genes that coordinate, orchestrate this delicate process, and even small change in the structure of these genes or on the regulation of these genes may have a profound impact on the spermatogenesis and the sperm production efficiency. And now I'm reaching the momentum. Over the last few years, almost less than a decade, we are now having in our hands powerful tools like whole genome sequencing, whole exome sequencing, genome-wide association studies, and RNA sequencing, that all of them provide us an amazing opportunity to further explore the genetic basis of non-obstructive azoospermia and try to find new ways of therapies, of diagnosis, and treatment, or new approaches for the genetic counseling for the people undergoing these treatments, for them to know how safe this approach is for the next generation. So we took advantage of this momentum, and we created a team, a powerful team, with EmbryoLab Fertility Clinic, EmbryoLab Academy, and the Thessaly University, the Department of Biochemistry and Biotechnology. So we combine a spectrum of knowledge and expertise that allow us to have one common mission, the mission of unraveling the genetic basis of male infertility for the Greek population. And this project, this huge research project, was named Spermogene. So in the context of Spermogene, we recruited for 4 years patients that were recruited through an awareness campaign. So we were able to register more than 1,800 patients. They all underwent a questionnaire and sperm analysis. We retrieved more than 1,200 blood samples from all of them, and we managed to gather 300 testicular samples, obviously after consents and everything. And the samples, blood and testicular samples and sperm samples, were stored in -20 before they were shipped to the university department. In particular, for the testicular samples, we did something that I think it's an innovation. We stratified the testicular tissue based on their quality in terms of the presence of spermatozoa. So we had the testicular sperm with totally absent of the presence of, with no spermatozoa. The testicular samples with high presence of spermatozoa, easily found immediately within 1 to 2 minutes. The average quality, where we had 5 or 10 minutes before we allocate the first spermatozoa, or the rare presence of spermatozoa, where it took more than 10 minutes to find 1 spermatozoa. So this kind of stratification of the testicular tissue gave us the opportunity to divide and explore the genetic basis in the different phenotypes of the testicular tissue retrieved from azoospermic men. And thanks to the questionnaire and all the tests we had available, we had lots of data on the population. So we had a well-described population, which is, as you already know, a very fundamental part for a good analysis and valid data. And this is the outline of what we've done. We recruited 26 patients that were either carriers of CF, which are diagnosed as obstructive azoospermia patients, and they were the ones that served as control. And for validation purposes, we divide and created 2 mirrors, 2 pairs of the same patient population, and the rest of the patients are coming from idiopathic azoospermic men. Men suffering, diagnosed with idiopathic non-obstructive azoospermia, and they were divided based into 4 different pools based on the phenotype. 2 of them were the ones with total absence of spermatozoa in the testicular sample, 1 pool with high presence of spermatozoa, and 1 pool with rare presence of spermatozoa. So for all these pools, they underwent RNA extraction, bulk RNA sequencing, and mRNA analysis, because we were aiming for protein coding genes. And then data stratification and differential gene expression was assessed, and we divided our genes based on the ones that were upregulated and the ones that were downregulated, and then proceed to bioinformatic analysis in order to understand how the different levels of spermatogenesis is coordinated. So again, a schematic outline. So all men recruitment with azoospermia were patients from Embryo Lab fertility clinic. They underwent a discussion, a counseling session. They had to sign a specific consent form before we proceed to the biopsy for in the context of the treatment, blood retrieval and questionnaire. Then all the samples were collected and transported in a safe protocol to the university where RNA extraction, RNA sequencing, and differential gene expression analysis took place. And what we have seen, outcomes. We have seen that in all of these tissues, different phenotypes, remember, with different presence of spermatozoa, from total absence of spermatozoa to almost normal spermatogenesis in case of our controls, obstructive azoospermia. We have thousands of active genes ranging from 27 to over 39,000 of genes. And when we are checking the protein coding genes, the range is between 17,000 to over 19,000 protein coding genes. In order to assess the differentially expressed genes, we created and proceed to the comparative transcriptomic analysis. We created pairs in order to check all the pools of inferior spermatogenesis towards pools with better spermatogenesis. And we came up with these graphs where in our volcano plots, these plots, we can see that when similar, these are CF obstructive azoospermia pairs, when they are compared, they are absolutely the same. So these serve as validation for us. Again, when we move to the Spearman correlation diagram, we have seen that the more similar the phenotypes, the higher the compatibility, the correlation according to this graph. But when we moved and we checked for differential expressing genes, we have seen again that the more similar the testicular tissue, the less the differences. But when we check and compare highly different testicular tissues, then the differential expressed genes is exploded and escalates to thousands of genes. And then we decided to see and check how many differential expressed genes were present in all our pairs. So there was over 3,800 differential expressed genes. And then we moved on to check which were exclusively upregulated and which were exclusively downregulated. And what we have seen is that they follow a pretty much distinct pattern where we have seen in the lower quality testicular samples, when the testicular, the spermatogenesis is severely impaired, we have a upregulation of certain genes and a downregulation of another group of genes, and only 131 exhibit mixed expression patterns. So we use the bioinformatic platform, the gene ontology analysis for differential expressed genes to explore which biological pathways, molecular function, biological processes, and cellular compartments were more enriched. And what we have seen was, in a way, expected, but what's interesting is the unexpected information. So we have seen that in good quality tissues, we have an overexpression of all the pathways that are linked to sexual differentiation, gamete generation, spermatogenesis, and etc. But what we have seen in the low quality testicular tissue is that there is an overexpression and increased expression of pathways that are not expected. These are involved pathways in immunodefense, in apoptosis, in regulation of cell death, in inflammation. And then we want to see which genes are mostly involved. And again, genes were downregulated, and genes that are well known that are involved in spermatogenesis, while the upregulated genes are well known to be involved in the regulation of inflammation, cell death, immune defense, etc. So we tried to explain that. And we went back to normal spermatogenesis. And what we have seen is that, and what we know, this is known thing, that spermatogenesis is only complete during puberty. So that means that male germ cells produce autoantigens and may trigger immune response. So this is why the evolution of the humankind has created the blood-testis barrier in order to protect and create a unique microenvironment where Sertoli cells play a very important role in maintaining, in protecting male germ cells against immune response. So they create a mechanical and physiological and as well as immunological barrier, a zone of suppressed immune response. And so autoimmune response is response against male germ cells is prevented. So this is very well known as testis immune privilege. So the testicular environment is well protected by the Sertoli cells. And the last thing we expect to see is the escalation of the immune-related molecular pathways, which is the case in non-obstructive azoospermia. So how can we expect the pattern and the transcriptomic signature we take from non-obstructive azoospermic PAN? It's only if we accept that we may have the failure of testis immune privilege, where the loss of blood-testis barrier take place. There is a depletion of the testis immune privilege, activation of immune system, inflammatory cell infiltration, apoptosis, germ cell depletion, and spermatogenesis eventually impairment. So thanks to this research project, which is ongoing, there is a wide publication, a wide range of data that some of them have already been published. And for the first time, they address the genetic basis of Greek population, which is so far was so far underrepresented. And this points out the need for intra-ethnic studies in order to validate the clear genetic basis of non-obstructive azoospermia and address ethnic variations. So our aim in our big and innovative approach is to move away from the genome to the genes and identify the genetic fingerprint of azoospermia and make sure with our contribution that we open the road or contribute to the new genetic approach, to new genetic approaches, new innovative treatment approaches, or the development of gene panels that will address non-obstructive azoospermia and the genetic basis on a personalized level. Thank you. Thank you.
Dr. Usha. Thank you so much, Dr. Alexia.
Thank you.
I would like the
Alexia.
experts to come. Yeah.
You would like us to comment?
Yes,
it was really a wonderful and advanced genomics, okay, for tackling and the management of NOA, which at the experimental laboratory level you have been offering to the patients and as a service, which is very good. What I would just like to just get, if you can highlight in your practice, whether you have developed some target panel genes, the gene panels, to very quickly detect this azoospermia, NOA, in clinical practice, because we'll be having both groups, and whether those who are very common and whether you are going to have, or whether you have, or in future, whether it's coming based on the testicular biopsies results. Yeah.
Yes, the idea is to develop a panel with all the genes, the biomarkers that have been identified that are associated with non-obstructive azoospermia, in an effort to have a valid diagnosis for non-obstructive azoospermia. We are pretty much aiming to identify the ones that have a negative biopsy outcome. So in order to relieve them from the need of a biopsy, as you may remember in the beginning of my lecture, I said that only approximately 50% of the men have a positive outcome, despite the efforts through the improvement of the testicular biopsy intervention and the processing of the testicular tissue. So only 50% will have a positive outcome, while the other 50% of men have undergone an unnecessary intervention. So the aim is to help these people from an unnecessary intervention. So the aim is to create or develop a panel that will address these men. But again, this is on one ethnic group. So even if we have this panel, I won't know whether this works for Indian men or for men from Northern Europe or USA. So this is where the collaboration is necessary.
Right.
So in order to test and validate these markers or other markers, in order to create a global as well as an ethnic-based panel, in order for it to be useful for clinical practices and clinical purposes, and for the urologists as well.
Yes, thank you. Thank you for that elaborative answer. Thank you. Is there any other questions? You are mute, Anuradha.
anybody has more questions? Can ask Dr. Alexia. Dr. Rahul, you have any questions?
I'm getting my laptop ready here.
Okay. So thank you, Dr. Alexia, for joining us today and giving your valuable insight into this azoospermia non-obstructive, the genetic basis. I think we are slowly actually unraveling these genetic problems, what we have in the male partners. And I think every 6 months, probably, we go to different conferences or we read papers, something new comes up. So probably what Dr. Usha is saying, that we are going to maybe develop a proper pattern. And of course, we have to keep in mind the ethnicity also, because genetics is slightly different in different ethnic groups, and how it unravels, we'll have to see. I think we have done a lot of, I think with genomic development and all, we have done a lot of advancement has happened, but I think we have to further, make it more and more available to everyone. That is how it is, and it has to be cheap. That is where the test has.
Very important point.
Yeah,
Very important.
It has to be
bring it in,
available in the
can
market.
I
It has
just
to be cheap.
comment? Because but for that, such collaborations are required. You, as the IVF specialist, should, or we from India, you should collaborate to establish lab, to get the techniques, give her, then only Dr. Alexia can see whether ethnic differences are how, and you can come out with the panel, specific to India. It's important.
Okay,
Yes.
so thank you so much. We move on to our stock two. That is again being done at a lot of places now. It started very small. It has become very huge now. So embryo biopsy, the newer perspective. And I would like to invite Dr. Rahul Sen, our experts, Dr. Ram Prakash and Dr. Ashwini GB. Dr. Ram Prakash is founder and scientific director of Mairi Fertility, Noida, Delhi. NCR, co-founder of scientific director at Angel Fertility Clinic, Nepal, is a visiting consultant embryologist at Max Super Specialty, Vaishali, Delhi, and certified clinical embryologist, has performed more than 10,000 IVF ICSI cycles and more than 1,500 biopsies. So I think you're the right person to be here as an expert, Dr. Ram. Welcome. And he has got more than 12 years experience. He's trained more hundreds of doctors. He's designed and implemented and executed IVF lab setups. And his keen interest is in embryo biopsy. So we are going to hear from him also. And our second expert is Dr. Ashwini GB. And she is my fellow Bangalorean. We are Kisarians. She's Secretary Kisar. Can I have her CV? She's the director of Aspire Fertility Center in Bangalore, and she's the secretary of ISAR. And she is basically an IVF specialist, and she does embryology also. So it's a two-in-one collab. And her special interest is in PGT, genetic testing, and all. That is why she is our second expert. Welcome, Dr. Ashwini. And Dr. Ram Prakash, could you please introduce our speaker, Dr. Rahul Sen?
Thank you. Thank you so much for the kind introduction. And I would like to thank you all for having me here. So our next topic for discussion is about newer in PGT. And presenter is my very good friend Rahul Sen. He is a great human, and he is a selected member for ISAR Youth Brigade for 2024 to 26. Past executive committee member for ACE, nominated mentor in the ASHRAE mentorship program for 22 to 23. He's also got many awards on his name, ISAR Shining Award at Embryology, then IFS Achievers Award. He was a past treasurer of ISAR Rajasthan chapter, past executive member of his IFS Rajasthan, and scientific committee member at IHERA. He is also invited guest faculty for multiple colleges, including Aurobindo University. He is in the field the last 15 years, and I know him personally from last 10 to 12 years. And he is also associate editor to ACE newsletter. He is a member of ISAR, IFS, ACE, ASHRAE. Over to you, Rahul.
Thank you so much, Ram, for this kind introduction. And on this onset, I would like to thank Dr. Anuradha ma'am, Dr. Seema Pandey ma'am for initiating this wonderful insight series, for not only sharing our knowledge, but also to communicate, to manage, to interact for our routine day-to-day problems what we face during our ART practices. And also thank to ACE national body for giving me this opportunity to interact with you all on this topic, which is very much close to an embryologist heart. When an embryo is ready to transfer, but we are not actually knowing about it, whether the embryo which we are going to transfer, that is basically a normal embryo or an abnormal embryo at towards the genetic point of view. So we'll be discussing about the embryo biopsy and its newer perspectives today. But I would like to take you back in the 90s, when these two guys, one is Dr. Robert Winston and other is Dr. Alan Handyside. they actually performed this first pre-implantation genetic diagnosis at London at Hammersmith Hospital for an X-linked disorder that was adrenoleukodystrophy disorder, and that was disorder related to an X-linked, and that could not happen if a female embryo is carrying those genes, and it eventually gave to birth to a healthy baby. And believe me, this road of biopsy, it was not started in 1990. This road was actually began in 1937, where the initial work was given by Dr. John Rock, and later on further work has been continued using mammalian embryos, and then coming back to the human embryos, human gametes, and then it came eventually for the birth of the first female that was in 1990. And this road not only developed from 1990 till now, it has given various advances not only in the technique as in term of the technology part also, whether we are doing a day 3 biopsy or a day 5 biopsy, which we'll be discussing in the later half of the presentation. It also has eventually given PGT a new name as a PGT. Initially, it was named as PGD or PGS as a part of diagnosis and screening, and then it eventually given a various versions of development where PGT 1.0, 2.0, 3.4, and 4.0, which is the recent, the non-invasive part. And this is how the PGT has progressed throughout its developmental phase, beginning with the fish of having a day 3 embryo, getting the single blastomere outside, and now after that it eventually went up with trophectoderm biopsy of using day 5 embryos, where array CGH, SNPs, and NGS was performed, and now it is a recent era which is of non-invasive PGT. But to basically to aim this whole process of PGT in a simple word is basically it is an exercise where where a genetic portion of an embryo is basically retrieved outside. It is retrieved and sent to the genetic lab for the assessment to evaluate whether the embryo is euploid, aneuploid, carrying any kind of genetic disorder or not. But our today's agenda is one and only embryo biopsy. We'll be discussing all the WH questions: when, why, what, how, where, all these types of questions related to embryo biopsy. So before beginning with the embryo biopsy, we need to set up our laboratories. We need to set up our IVF laboratories, and that setup basically doesn't require any kind of specialization. But yes, there are few devices like laser system which needs to be incorporated in your routine micro manipulator system having an inverted microscope in it. So laser system is basically a need for an invasive PGT process. And not only with the laser system, I am sure my experts will also agree on this fact that there is a mandatory requirement of having an proper anti-vibration table. We cannot go with the simple routine tables which we are getting for our routine ICSIs, but yes, there is a requirement for a specialized anti-vibration table, and there are the few consumables and culture media requirement is also there. So for next few slides, I'll be basically covering the embryo biopsy part and what stage of embryo we should biopsy, what time we should biopsy, and there are the techniques that basically the process of biopsy can be done with. So we all know that day 3 biopsy was an earlier era, which was eventually started with the PGT program, where a day 3 embryo was basically lysed, the zona pellucida was drilled, and one single blastomere was taken out and sent for the process of genetic assessment. But with the later era, with the later onset, and with the advancement of the PGT, it came with the fact that why not to undergo blastocyst biopsies? Why we are restricted only for the day 3 biopsies? So it came with the fact that yes, blastocyst is more robust, more developed, having specialized kind of cells, having specialized divisions, like with the series of progression, a blastocyst basically develops into a specific cells like inner cell mass, trophectoderms, and there is an expanded cavity. So why not to undergo a blastocyst biopsy as compared to day 3 biopsy? And later, with this eventual phenomena, it came with the fact that the blastocyst biopsy remains a gold standard of practice as of our current era of PGT practice. Blastocysts basically have more robustness, more specific cells of formation like inner cell mass and trophectoderm cells, and it provides huge spectrum of array to provide more DNA that can be eventually amplified, analyzed, and can be interpreted. And it has also the lesser chance, the lower phenomena of having mosaicism with the good implantation rate as compared to the day 3 programs, which we used to eventually do in our routine earlier practices. Now, I think it is basically a gold standard practice for an blastocyst biopsy to undergo PGT. So, as we were discussing that the blastocyst formation is basically a natural phenomenon, a physiological process where after the modular formation, the blastocyst basically forms three to four different stages like early blastocyst, an expanded blastocyst, hatching blast, and completely hatched blast. So we need to understand the physiology also, and we need to understand the developmental phases also. Like in the early developmental phases where the blastocyst is not having a good preformed cavity, those cells cannot be, those blastocyst I think cannot be considered as what we practice in our routine practice in embryology. Like these blastocysts are too premature to perform biopsy, and from these blastocysts we may get the insufficient trophectoderm cells, or we may be damaging those inner cell mass also, uh, also for biopsy. But if we undergo biopsy in expanded cells which are well developed, they are having very good trophectoderm, they have intact zona or the inner cell mass. So these blastocysts can be have a good option. They can be considered as a better option for the process of biopsy. And we all know this fact also that not always we can actually aim, or we can actually look for always having a good quality blast like a 3AA, 3AB, 6A, 6AB, or BB kind of blast, which we considered as a good quality blast. There can be eventually evidences, or there can be eventually conditions where we can actually look into these kind of facts also, that we may be having a compromised, or we may be having some poor quality blastocyst available for biopsy because there are no other options available with us. So there can be a mixed group of blastocyst formation which we can actually get out of an IVF cycle. So we need to actually grade them properly. We need to actually grade those blastocysts, good quality blastocysts properly. We need to circle them we need to culture them separately, so that we are actually doing biopsy on majorly on the good quality blast as compared to the poor quality blast. So ideal choice, what we actually practice and what is actually recommended as an good expanded blastocyst, at least with a 4 or a 5 grade of expansion, with clearly demarked, with clearly visible trophectoderm cells, and a proper recognized and a proper seen inner cell mass. So with the technique, there are the two techniques which are there. One is the technique while we can do a biopsy using a stretch method with the help of laser, and other technique which is basically a flick method with the help of laser. So there are these two techniques. I got this wonderful picture from the Cooper Surgicals, and this is how the process of biopsy technique.
So we're positioning the embryo as
Yeah, just yeah. So, so this biopsy technique basically works with the help of laser, and the stretch method is there. And this method of biopsy can be more significant, more be viable for those embryos which are actually hatched out of the zona, and they have this zona opening out of this proper hatching blastocyst out of it. After the collapse, with the proper suction, with the proper handling, and in the horizontal plane, this embryo biopsy can be performed using multiple laser shots. We need to be more careful with the laser shots, because we should not damage the trophectoderm cells. The aim is to get the good quality trophectoderm cells without damaging the remaining part of the embryo. And after the tissue is being slashed out, it can be flushed and relocated on a different media platform. And the other technique, which is the flick method, using using the laser technique. And this technique basically functions. I have two different videos showing. One is showing an uh, intact zona with the with the blastocyst inside it, and other is showing a brief portion of trophectoderm which is being hatched out, and again getting into the horizontal plane, aspirating few cells, giving it a laser shot, not more than 2 or 3 times. The laser shot can again the intensity of laser shot can again be reduced. And soon after the biopsy, soon after the flicking method, that flicking method can be done using fixing the holding pipette and getting the biopsy needle using this that causing a flick using the holding pipette. So with this technique of biopsy, laser and flick can be performed. And we need to also understand this phenomena, that we should not take more than 5 to 10 cells. We should not take more than 10 cells, because ideally we require 5 to 10 cells that should have sufficient amount of DNA that is reliable for the genetic analysis, doing no minimal harm to the remaining blastocyst part, which is there for the they're available for the transfer or cryopreservation. Aim is to clean, get the control biopsy out of it, get the cohesive trophectoderm cells with the proper appearance, and enough material is accurate for the diagnosis without minimal damaging. And in few cases, like the embryo, the blastocyst, which is having a less developmental part, or the less trophectoderm, in those portions, laser assisted hatching can work. Laser assisted hatching can be done on day 3, day 4, even on early blastocyst of even on early day 5 also. That can after that we can actually perform a biopsy, a late biopsy on day 5, that can eventually help us in getting improved biopsy, improved efficiency in biopsy, reduced embryonic manipulations, better yield of trophectoderm cells, and again there could be less chance of giving minimal thermal shots of laser to prick to get the trophectoderm cells out of that developing blastocyst. So after getting the biopsy part, the biopsy cells, the trophectoderm cells, we need to eventually analyze those cells also. We need to we can also grade them, we can also analyze them by their cellular integrity, by the number of cells present, by the cohesive properties, by the cytoplasmic appearance. So we can actually we can actually means we can intellectually say this was the proper cell, this was the proper trophectodermal cells which was biopsied, and later it were sent for the genetic assessment. Better to take pictures, better to take recordings, so that we can on the later part also, even after the amplification and the interpretation of the report also, we can go back and see how these trophectoderm cells were there, whether they were having a sufficient amount of DNA or not, whether they were having a poor DNA profile, a report of poor DNA profile, so that we can actually go back and do the troubleshooting. And there can be few scenarios also where we can get into the trouble part, that our biopsies, what we have done, these reports may have suggested that there were the insufficient trophectoderm cells, there were the lies or the fragile trophectoderm cells which we have got out of it, there was the difficult trophectoderm aspiration, where we were not using less or the more laser part. The embryo biopsy, we may have got the more cells. Zona pellucida was not, means it was more over the sticky or the thick part was there. So to means to take care of the troubleshooting part, we need to go back when this IVF cycle was about to start. We need to go back to that starting point of oocyte retrieval on that particular day. How were our equipments working? How were our laboratory conditions were working? How our culture conditions were working? How was the QC QA of the lab? So all these things will play a major and the most important part in the embryo biopsy on the day of embryo biopsy, because we have done the fertilization part, we have done the culture part, and the embryo has now turned into a blastocyst. And throughout all these 5 days, how were the lab was performing, how was the culture condition was performing, how were your equipments were performing, all these things plays a major role. So in few conditions, there can be conditions like we need to undergo a rebiopsy. In those conditions, we need to recalculate few things, like the word rebiopsy, post biopsy, it itself, as what we in our practice we have done this abbreviation, that rebiopsy itself should be claimed as why there was a repeat of biopsy that needs to be considered. What was the evaluation in the initial result? What was the blastocyst quality at the time of biopsy, at the time of formation, and at the time of free biopsy? Whether we have done informed consent to a couple, whether the optimal number of trophectoderm cells were removed now after the rebiopsy part, whether the handling, whether the culture conditions, QA, QC, everything, it was precisely controlled. Strict cryopreservation protocols needs to be followed. And yes, the aim again, it should be the same, whether the yield of DNA, it was it was enough, it was sufficient after the rebiopsy part. So the rebiopsy can be considered in few cells, in few conditions, and in conditions like an embryo is having partial hatching, like this type of blastocyst is having a partial hatching, and in this condition there are the few cherry kind of cells which are actually hatching out, which are extruding out of the zona. So do we need to pick these kind of cherry cells? What we routinely say in our practices, basically it is a cherry picking portion. So this cherry picking can be avoided, this can be excluded when these cells are seen, or they are look like a degenerative fragmented part, loosely attached part, they are not having a good cohesive kind of appearance, the cytoplasmic appearance is onto the darker part. So these cherry picking kind of biopsies can be avoided. And after the biopsy part, there comes the again an important aspect, whether to how to tube and how to load those biopsied cell into the PCR tube that can be sent to the genetic lab for the genetic assessment. It is basically what we routinely practice is to get those PCR tubes in a in the edge of the petri dish, and that basically grasp that can basically have this adherence property. It will not move, or it will not relocate, it will not rotate also. So in that condition, we can have this proper appearance of the bottom part of the PCR tube also, and in that particular zone we can actually relocate, we can actually place the biopsied material, and using proper dish preparation techniques, we can actually rinse our micropipettes with having less contamination of oil, any kind of culture media, any kind of other cellular remnant. So all these techniques can be properly managed with the help of proper with the help of proper laboratory processing, and these kind of technique will eventually have a good interpretation, because it will not have any extra DNA material, it will not have any kind of extra contamination in the report. So we need to uh, uh, properly maintain an SOP, properly maintain a chart for the tubing and the loading portion of the biopsy technique. After the process is being done, we need to focus on what are the key performance indicators that we actually aim for, that we actually should look for. How an embryologist is basically being performing with this biopsy, how we can monitor that particular embryologist, what is the quality of the performance of an embryologist. So as mentioned in Vienna itself, there is a biopsy success rate of 90 to 95%, but itself we can from our own side, we can from our own set of conditions, we can actually claim few key performance indicators, like the post biopsy survival rate of the trophectoderm cells, the what was the amplification report, was what was the interpretation, and how was the interpretation being followed up after the process of biopsies. So there can be there can be many factors that can influence PGT result, what and how, what where was this technique of biopsy was being performed, whether there was any mosaicism, whether there was any contamination, how was the operator skill, what was the lab and laboratory and culture conditions during that period of time. So to conclude, yes, we can consider this technique as it is a technique of precision today, and a huge potential for tomorrow. Yes, embryo biopsy is a critical step. Day 5 remains the gold standard, and the better technique for the better yield of DNA, and with the minimal impact on the embryo viability. It is a proper technique for the handling for the tube. It is the proper technique which can be followed and replicated. There is again a need of monitoring KPI in terms of biopsy success, amplification, and embryo survival post biopsy, post cryopreservation also. And with these standardization and quality control, yes, biopsy can be performed safely, and enhancing success rates and patient outcomes for IVF. And not only with this invasive part, there is a new paradigm which is rising, and it is rising in a particular fashion, that there is now no invasiveness needed for this genetic assessment. There can be few conditions where culture media can be evaluated, and few portions where blastocele fluid can be evaluated for the genetic assessment of an embryo. But this non-invasive technique remains very naive, very silent approachable technique, and it is yet a technique that needs more exploration, that needs more conclusive data to evaluate and to use it for the commercial purpose. Thank you so much.
Dr. Ram?
Thank you, Rahul, for amazing presentation, starting from what are the embryos to biopsy, to set up for embryo biopsy. Then troubleshooting is one of the important part while we perform embryo biopsy. And also another thing which you highlighted was the most important is rebiopsy. In many cases, we all have these problems that the genetic lab reports us. They tell us that sufficient sample was not there. Then we again have to go and rebiopsy. So what are the factors are there that also you have mentioned beautifully? And the KPIs about embryo biopsy is one of the most important part to get consistent results for your patients and to get consistent reports for your embryos which you have biopsied. And ultimately, whenever we all perform embryo biopsy, our main goal or aim is to preserve that embryo, to give that viable pregnancy. The goal is not embryo biopsy or to get the report. The goal is to preserve the potential of that embryo and deliver into a healthy baby or deliver into a baby. So one thing, Rahul, I would like to ask you, like for the people who are planning to set up new embryo biopsy program in their existing IVF clinics. So what are the checklist does they require before they start the embryo biopsy program at their clinic?
I think the first and the foremost thing which they require is the checking with the locations of the genetic lab, whether they are very much readily approachable to the genetic lab or not, whether the distance between the genetic lab is nearer or it is more far away to their means what is the accessibility of genetic lab. And even after that, they need to check how their culture conditions, how their embryo culture program is working, how is their blastulation rate.
Palak,
They need to again
can you just put the
get
volume for
good
this one
culture
now?
conditions. They need to focus more on good
It's the volume
blastulation
button.
rate. They need to purchase few of the equipment like with the existing ICSI machines like micro manipulators and inverted microscope. They need to have at least a good laser system. I think the other disposables and the culture media part can be taken care of easily. But yes, from my perspective, I think the approachability to lab is a major concern which one should look
It's
for.
very, very important. Yeah, absolutely. And I mean, like after biopsy, how you keep your sample is very, very important. I know in many labs we have seen the people they do biopsy, they leave sample at the normal refrigerator, which should not
Yes.
ideally happen. So these are the few checklists. I mean, so again, one of the most important part which Rahul have mentioned that before you start your program, you have to make sure that you are equipped well.
Palak,
You have all the things placed in your lab, including your freezing, cryopreservation. Apart from that, I mean, like what I'm talking about freezing is the your refrigerator where you store your biopsy samples and access. As Rahul rightly mentioned, that your samples should not be at your lab for longer time. You should reach to the genetic laboratory as soon as possible. And also, Rahul, while we talk about like as we have a lot of fellow embryologists who are interested in learning embryo biopsies and who are practicing for this. So, what should be the minimum embryo biopsy a person should do before doing to an actual patient or to performing to actual a patient's embryo? So, according to what should be the minimum number?
Yes, I think we all have discussed this since long ago that before giving any embryologist a training for biopsy, we need to look back again and we need to understand how that embryologist was using micro manipulations for ICSI or some other techniques. So we need to understand what is their FERT rate, what is their good blastulation rate out of performing ICSI using the same micro manipulation system. So after that, we can actually think that the particular embryologist is now suitable candidate for learning biopsy. And at least I think minimum of 100 to 150 blastocyst to biopsy. I think those should be the minimum number because one need to again understand.
Absolutely, that should be minimum.
Yeah. Yeah.
Yes, it's a basically it's a more of a
May
I come in?
skill game, and also at the
Doctor,
same time,
May I come in, Rahul?
and
no, one second.
apart from this, also the person. Yeah, Sanket, do you want to mention anything? The
Yeah,
person,
Ram.
whoever is
After you.
planning to do biopsy, they should get themselves validated through a genetic lab on the practicing embryos. Yeah, Sanket, you want to add something?
Yeah, Ram. Thanks. Excellent presentation, Rahul. And question to Rahul is Rahul, can you please touch, maybe perhaps for the younger audience who has just started doing biopsies and getting validated, could you please also mention about the angle of the pipette holding and also the biopsy pipette? Because at times I've seen people keeping at 35-degree angle and they're practicing with 30-degree wala pipette. So
Yes.
how does it impact on the embryo outcome as such?
Yes, that is what we were discussing, that an embryologist should be well explored, should be well aware of that particular micro manipulation system, whether the
Correct.
micro manipulation system is working fine or not, whether they have performed ICSIs on the same micro manipulation system, what were their outcomes of micro manipulation like after ICSI, so that an embryologist is not only well aware of the machine also, but they should be well aware of the technique that they are actually going to handle a blastocyst, and they will be performing a biopsy out of it. So they need to understand both part, the machine part and the technique part as well.
Correct.
Yeah.
Anu, you're muted.
Rahul, I think
Dr.
Pratik also
Ashwini is
mentioned
there. One second,
mentioned
Dr.
it.
Dr. Ashwini is joined in.
Hi
Uh,
guys, thank you
so-
so much.
So you please, you have to
Yeah.
say anything.
A little something, Rahul. I was not able to attend the whole thing, but I had an emergency came in between. But excellent presentation. Really happy that I joined and I'm amidst all this elite group. Rahul, one additional point. That's all. Would there be a difference when a biopsy is done for aneuploidy detection versus mosaicism and structural rearrangement detection? If so, what additional care would you take for PGTM or PGTSR, or is it the same?
No, I think it won't be the same. Again, I'll go back to my presentation and reflect that same point, that the process of biopsy, it doesn't start on the day of biopsy when we are performing this day 5 blastocyst biopsy. We need to go back again. We need to check what were the laboratory conditions. We need to go back and check what were the the culture conditions where the embryos were growing. We need to go back how the process of fertilization which was done, because mosaicism and structural rearrangements, these are not these are not the part that
No,
actually
not
a biopsy.
mosaicism, Rahul. For
Yeah.
mutation. Sorry, maybe I worded it wrong.
Okay.
When you're doing a biopsy for PGTM, that is, you're wanting to check for a mutation.
Yeah.
So, what is the additional caution that you might take?
Yeah, for the PGTM part, I think it is again a necessary understanding that an aneuploidy should be evaluated first, whether after going for a PGTM for the same embryo. So we need to rule out, we need to check there should not be any kind of aneuploidies other than the consisting mutation which is there in an embryo. So we need to rule out that
And
first.
one more
Then
thing
You
Yeah,
might
to
want to
add to
change
that is
everything.
Yeah, yeah.
For every
Yes,
embryo,
yes.
you might
yeah.
want to change.
Yeah. Can I
Yeah,
add something?
for
Yeah,
the
Rahul.
PGTA, we can, I mean, like we can do biopsy only to the embryo which are best, but for the PGTM and for PGTSR, we need to biopsy every possible embryo because those are the very special cases in that.
Correct. Correct. Just to
Dr.
add
Pratik
what
has one. Dr.
just to add
Sanket.
what Rahul and Rama said, yeah, the only part is the biopsy wise, it remains the same. We as such don't see any difference because the panels which are set at genetic lab, first they do aneuploidy screening, followed by your PGT-M, whichever monogenic disease that is. So I think the panels are set in such a way that they has to pass through aneuploidy first, then we will go to your PGT-M or SR.
True.
There's a question in the chat box quickly. As we all know, recently there was a statement by ISAR IFSAs ruling out NIPGT to be used as a diagnostic test. It came few days back. So what is your take on it? And Pradeep, can you please come in? You are there, I know. Okay.
So
Okay, Dr. Ram
I
or
think-
anyone can answer.
yeah, I think in
The joint statement which came.
in the same slide, I think the last slide which I covered of the non-invasive part, and this non-invasive technique is yet still not a decade ago, means it is not a decade old technique. It is again a younger technique that needs more evaluation, that need more validation, that needs more data out of it. I don't think there is some conclusive evidences which are there in favor for this technique of non-invasive assessment, because again this technique is evaluating only aneuploidies. This technique is not conclusive in terms of monogenic disorders or any kind of known disorders which are there with the couple. So yes, this technique is not yet having much favorable conditions to perform in the routine ART practice.
So the take-home message we are giving on this platform today is that we are going to stop doing NIPGT. Dr. Ram?
No, but what does the statement say?
societies
Are there
all
any
have come
concordance
up
levels
with
that are matching with invasive biopsy?
Not yet.
Not yet, right? I think myself and Rahul had a debate on the same
Maybe it's a
Yeah.
long,
couple of
It's a
years
long
yeah,
ago.
maybe it's a
right
long way,
now.
way,
Be
but
continuing or
not
we are
right now.
stopping it.
Yeah,
Not right now. Maybe in future it may come again. Like
correct.
how earlier
So
we
right
saw
now the
proteomics and
take-home message
metabolomics
is
were in play. Then when
They were claiming 85%, 90%, but I think we were a little far away from that.
And
so
there
right
are
now the
only
take-home message is that NIPGT should not be used as a diagnostic test right now because all the three important societies, ACE, IFS, SRS, issued this statement. Thank you so much, Dr. Rahul, Dr. Ashwini,
Thank you so
Dr.
much.
Ram. I wish we had more
Thank
time, but we
you.
have to move on to the panel. Thank you for joining. Our next
thank
panel,
you so much.
next in line is panel discussion. That is
Thank
our
you.
favorite topic of clinicians, embryologists, troubleshooting in IVF. And I would like to invite our experts, Dr. Charulata Chatterjee, Dr. Raki Singh, and Dr. Charulata is our management committee member 22-25, and she has been past executive member of ISAR, IFS past joint secretary Telangana sector chapter, and she is scientific head and consultant embryology Medsi IVF. She has had a lot of paper presentations and she attends a lot of conferences, and she has like ESHRE SRM and ISAR Guru Award and ISSRF Excellence Award. And she is more than anything, she's a wonderful person and excellent embryologist. Welcome, ma'am. Our second expert is Dr. Raki Singh. She's president of Noida Society, Governor Council member of ICOG 24-25, vice chairperson of UP State ISA, Pear River of Jogi, and chairperson endocrinology. She's been past chairperson endocrinology committee 22-23. She's director Abalone Clinic, maternity and fertility and IVF center in Noida. And she has a lot of awards, and she's been invited as faculty for national and international conferences. But she has Foxy UNICEF. She's a Foxy UNICEF certified trainer for violence against women and cancer prevention. And she's going for elections. All the best, Dr. Raki, for Foxy Vice President North 2025. And our moderators for the day, my favorite people, Dr. Nishad Chimote, Dr. Manisha Vajpayee. Okay, Dr. Nishad Chimote. Dr. Nishads. Can you display the CV of Dr. Nishad, please? Yeah, he is fellowship of FICRM, director of Vasundhara Fertility Center, founder of Embryo Lab Evolution, second vice chairperson at present Embryology ISAR, Reproductive Genetics Working Committee, Aspire, executive committee member ACE 22-26, scientific committee member IHERA 24, joint secretary IFS Vidarbha. He's just finished that stint. More than 2 decades of experience. He's got a lot of awards. ISAR Excellence Award, ISAR Award, Champions of ISAR Award, Best Debater 22. And he's interested in lab designing and total quality management, AI in embryology, PGT, blastocyst culture. He's got 21 publications, international and national. Welcome, Dr. Nishad. Our second moderator is Dr. Manisha Vajpayee, a professor doctor. She's head of Department of Embryology and Research, Pacific Medical College and Hospital, Pacific Medical University, Udaipur. She's the managing committee, ISAR, executive committee member is executive committee ISAR Rajasthan, 23 peer-reviewed international publications in index journals. And she has presented original research paper at ESHRE and Aspire. And she has the first international publication from country on infertility and microbiome, ISAR Shining Star Award, ISAR Embryology Award, IFS Award, and there's no end to it. Welcome, Dr. Manisha. And I would like Dr. Manisha, is she there? Yeah. Dr. Nishad, Manisha, could you Could you please invite your panelists?
Okay, ma'am, I'll do that. And please help me with their CVs. Thank you so much for having us here. It was wonderful to be a part of such a prestigious and very practical series. So to begin this panel, I would like to invite Dr. Akash Agarwal. Dr. Akash Agarwal is MBBS ASHRAE certified clinical embryologist. He was topper, masters in human reproduction and embryology. He is heading QA/QC embryology at Nova IVF. He is managing committee member of ACE India 22 to 26 and scientific committee member of IHRA, mentor to CHFE, IIT Hyderabad. He is assistant professor guest faculty at Aspire University of Mysore. He is an alumnus of Osmania Medical College Hyderabad, topper of ASHRAE Clinical Embryology Vienna 2019, and invited faculty to multiple national and international conferences. I welcome you, Dr. Akash. Our next panelist is Dr. Brijesh Upadhyay. Dr. Brijesh is PhD and MSc in clinical embryology. He has done PGD genetic counseling. He is expert of NGS and PGS. He has done BSc in radiology and imaging diagnostic. He is having his keen interest in ART, QC, QA, ethics, medico-legal expert. He is ISAR, ASHRAE IFS certified embryologist, multiple paper posters. He has presented in multiple national and international conferences and journals. And he is having more than 5 years of experience in IVF sector. So I welcome you, Dr. Brijesh Upadhyay. Our next panelist is Sanket Dhumal. All of them are very good friends. Dr. Sanket is a ASHRAE certified clinical embryologist. He is core committee and scientific committee member of IHRA. He is member of FIDES World Health Organization. He is on advisory board of Spaceborne United. He is a host and mentor of IVF Tribe. He has done his MSc in genetics and MSc in clinical embryology from University of Mysore. He has done his PhD. His PhD area of specialization was reproductive biology. He is Hall of Fame embryologist of Economic Time Health World 2023 in Best Embryologist and many other accolades. He was executive committee member of ACE from 2019 to 24. He is editorial member of many prestigious journals. So I welcome you, Sanket. Hope you are there.
I would like to introduce myself. She's a good friend of mine. We trained together with Dr. Kamini. So I welcome Dr. Jyoti Bali, Director, Baby Soon Fertility and IVF Center. She has done her fellowship reproductive medicine along with me. She is president of DGF Central, founder secretary Delhi Gynec Forum, secretary Delhi ISAR 2019-20, joint secretary National IMA Women Wing, recipient of Nav Shakti and 8th National Women Excellence Award, Pride of India Award, and Dr. AP Abul Kalam Appreciation Award from Delhi Forum. And Economic Times has said that Inspiring Gynecologist of North India Award. She has got it. And welcome, Dr. Jyoti. Our next is Dr. Manjunath. He is not sent his CV, so that's why I said I'll introduce. Dr. Manjunath is our past president of Karnataka chapter of ISAR, and he's a very young and dynamic, and he is one of the national coordinators for Birla IVF Center. And he is basically an IVF specialist, but more than that, he's a good laparoscopic surgeon and a good friend of ours. And the next one. Yeah, Manisha.
Oh, so I would like to welcome Dr. Deepti Rapalwar. She is currently working as lab director in Mohak High-Tech Super Specialty Hospital, Indore. She is assistant professor and head of the clinical embryology at Sri Aurobindo University, Indore. She is having more than 18 years of experience as an embryologist in India and abroad. She is teaching faculty for DM, MCH and reproductive medicine in Sri Aurobindo Institute of Medical Sciences. She is member of ISAR, ACE, IFS. Her area of research are sperm DNA damage. Her area of interest in oocyte vitrification and embryo biology. And she has also presented various papers and posters in various prestigious conferences and published many papers in peer-reviewed journal. So I welcome you, Dr. Deepti. May I have the next CV?
No, I think we are done. We are done, and we can start the slides now. Slideshow.
Okay. Nishad, are you sharing the screen?
Yeah, I'll be sharing.
So at the onset, I would like to congratulate and convey my heartfelt gratitude to Anuradha Khar ma'am and Seema ma'am for initiating this wonderful series and for initiating this wonderful discussion as something very, very practical, which can be, which should be discussed by every ART professional, because we all know to err is human, a phrase we have all heard. But in IVF setup, it carries an entirely different weight. In this setting, even the smallest deviation can alter the course of someone's future. May I have the next slide, Nishant?
Yeah, please do so.
So, as they say, if there are two ways of doing something and one leads to a catastrophic outcome, sooner or later, somebody will choose that path, not intentionally, but because humans are fallible, systems are imperfect, and stress is real. But the beauty of a good system lies not in just precision, but in preparation. In our field, we don't just strive for success. We are constantly on guard against the failures. And when failure is threatened to creep in mid-cycle in real time, the true skill of an specialist or embryologist is how quickly we recognize the problem, how calmly we respond, and how effectively we prevent it from happening again. So in today's panel, we bring you the real-world troubleshooting scenarios from the embryology lab or IVF OT, cases that could happen any day to any of us. We will walk through them together, asking what went wrong, what could be done better, and what system can we build to make sure we catch the error before it reaches to the embryo. So let's begin. And before onset, I would like to request Charulata ma'am is already there as an expert. I would like to request Prateek and Ram to share experiences, and Rahul as well to share their experiences. Over to you, Nishant. Would you like to take up the first question?
Yes, so I think you've done a wonderful job with the introduction. I think that is what troubleshooting is all about. And glad to have such an arsenal of panelists and experts. And I think most of our doubts will be cleared. The questions are quite basic, but quite serious. What we need to do, what is required, because what can be done for troubleshooting is what we're looking for. So it is basically total quality management that has got so many nuances, that is from encompassing the training. The most important is SOP, proper communication, feedback, witnessing, and obviously good backup system. So basically, working in an embryology lab is like walking on a tightrope. Even a slight change and you will fall. And that is what we expect the embryo culture and the development of the embryo and the transfer and implantation at the end from the lab end. This combined with the problems when we face with the patients, especially the clinical side, the protocols, the types of gamete, the age of patients, these are all the things that can cause a problem. And that is how we come to our first question. So let me begin here. What steps should be taken in the lab when we retrieve the oocytes are predominantly immature? That is, you are getting only GVs and M1s. There are no M2s. And when retrieval needs no oocyte, that is another case. So you can answer on two fronts. So for example, how would you verify an ovulation trigger issue and attempt to rescue, such as a delay in repeated aspiration by a few hours? Would you culture immature oocytes overnight to see if they reach M2 for late ICSI, that is called rescue ICSI, or what are the success considerations? If no oocytes are obtained, how do you distinguish between true empty follicle syndrome and correctable error? So I would like to start with our clinicians here, Dr. Jyoti and Dr. Manjunath. How would you handle this saying that you're doing the pickup and there are no oocytes in the first few follicular fluids? Yeah, Dr. Manjunath.
Yeah, good evening, everybody. See, first of all, when we don't get the eggs in first few follicles, I usually stop and check whether the trigger was given properly by doing a follicular fluid test, HCG, the KIT test. If it is positive, then mostly trigger is not a problem. But if we have
Yeah.
given ANTAG injection, we can't do that.
Exactly.
So especially when we give an ANTAG injection, in case of some of the patients, like those who are put them on like long-term OC pills, there is over-suppression. The LH is not proper. So in those cases, this can happen. So in those ANTAG cases, we usually re-trigger and do the pickup after 34, 35 hours. Most of the cases, we have got the eggs in when we do a re-trigger or whatever the rescue HCG, whatever we give. So this is what we do when there are no follicles, this thing.
All right. Okay. Dr. Jyoti, what is your take on this?
See, we have a protocol in the unit that between the pickup day and the trigger day, there's a day in between.
Yeah.
So we always ask the patient to send us a message on a particular phone number at that particular time when the trigger has been taken. So that remains as a proof. We don't need to cross-check while in the OT because you're absolutely sure on the day of pickup that the trigger has been taken. Then there have to be other causes for it. It can't be a trigger timing which has gone wrong. That can be easily avoided because there's a day in between the trigger and the pickup. So that's a standard protocol that we follow for all patients, that if there's a mistake which has happened, you should know before the pickup. In between day, you will know about it. So you correct it and then post the patient again. Give an ANTAG, give a trigger again, and then reschedule the pickup. But if at all OT cross-checking has to be done, one can cross-check in the OT. After this would be just a formality to check in the OT. And not then as Dr. Manjunath said, of course, reposting can be done after 35 to 36 hours if there's an error. We even pick up the phone and talk to the patient after the message also that yes, it has been correctly administered or not. And we ensure that it has been correctly administered. If nobody is there to help her or she's not confident, you would know during the 12 days injections as to how she's taken herself or whether she was taking help. If there's a help to be provided, we even arrange for that help for the trigger that she can visit the hospital and take it. We'll arrange for a nurse or whatever. So this is how this will be troubleshooting can be done here.
Great, great. That's a wonderful answer. Now moving on to if you are getting GVs, so and advance. Now is a time that
you are not audible. I think your volume is a little lower. Please increase the volume.
Can you hear me now?
Yeah.
Yeah. Okay. Right. So now in case that you're only getting GVs and M1s. So in such a case, this question is slightly now towards the embryologist, that would you go in for rescue or would you go in for IVM? That is the rescue IVM, because IVM is totally different, I believe. What I want to say is whether you go for IVM, whether you culture the embryos, then do the ICSI, is it feasible and will it yield any result? I can start with Deepti here.
Deepti?
Yes, good evening.
Yeah.
If we see only GVs and M1 in only particular patient, then we might be say that might be the patient factor. Or if you are continuously getting for 3-4 patients every day, like GVs and M1s are less M2s, then we have to go back and see the clinical protocols also. If it is M1, then definitely we'll culture them till they become M2 and then we'll go ahead with ICSI. In case of GV scenarios, they probably take more than 24 hours to get into M2 and the chances are little less.
Hello.
Yeah, so in such a case that you're saying that it takes 24 hours for the GVs to get into M2s, but is it worth it doing the ICSI of such oocytes and then you expect maybe a good day 3 embryo or maybe a good blastocyst? Do you think it works?
I personally don't think they'll work, but when we don't have any other option, at least we have that option to culture and we'll see. That is the least available option we have, and it depends on the patient scenario.
So what is your protocol? If you are getting more of GV, are you keeping them for 24 hours and denuding them after 24 hours? Or it is, do you have any defined time limit? Okay, I'll wait till this time. And for M1s to M2s, do you like to wait? And if yes, for how long?
For denudation, we'll definitely do after 2 hours. Maximum 2 hours we'll keep the incubation. That is our routine protocol. After pickup, we'll wait for 2 hours, then we'll go with the denudation. So rarely we'll see all the oocytes might be GV or M1. If they're M1, then we usually wait for 3-4 hours. If all are M1, if some are 1 or 2 are M1, then we don't wait for the M1s, we'll go ahead with the M2s. If there are GVs, then we have to wait, that which is a very rare condition. And then we'll see and we'll decide, we'll counsel, probably we'll inform the clinician. After counseling, they'll decide whether to go ahead with ICSI or whether they'll go ahead with another cycle. Or we'll also see the clinical protocols, whether they have any previous pickup where they have also seen the GVs, or only this time, then we'll take on the action what to go ahead or to do.
Anybody else would like to add something? Sanket, Akash?
Yeah, thanks, Manisha. Think, just to extend what already have been discussed. So once clinical part is done, so since we are not going for standard IVM as such, it is as Nisha rightly said, it's a rescue IVM. So just to salvage the situation, I think, personally in 13 years of my experience, I think I've never seen, say for example, more than 5 or 10, you're getting that, and hardly you see all are becoming GVs or M1s. Very rare. Mostly this happens with the lesser follicular count, say when you're getting less than 5 oocytes, the chances will be more. So in that cases, I think, just to salvage the thing, I think culturing them for till next day. If the pickup is done early in the morning, say you've done it after 2 or 3 hours of the pickup, or say the 38th hour of post HCG. So then, if you find such gametes there which are not mature, so I think culture them in the same culture media, since we don't have a backup as a IVM media as such. So check back in the evening. If they are mature enough, I think their vesicle has been broken down, they're converted into M2s. Then you can do ICSI on the same day itself. Otherwise, last but not least, I think we have to go for overnight culture. But the conversions are very less, I would say, not compared to the standard conversions what we get for day 3 as well as day 5. So that has to be taken into consideration, and I think counsel a patient accordingly.
Okay.
Right. So basically, waiting for 24 hours for the GVs to get into M2s and then doing, it's just hoping against hope and a mere formality. So this is the basic message to our audience, that yes, you can do it, but it will not, you do not hope that it is going to get converted into a blastocyst and maybe you'll get a good pregnancy out of it. It is just hoping against hope, and we try our level best in the lab to do whatever is possible. Yet we know what the outcome will be. So that is one thing. Also, second is about the trigger, that around 6 to 12 hours, this was the paper by Lao Zhu et al., that post-corrected trigger of 6 to 12 hours of when there is so-called empty follicle syndrome has yielded good oocytes. I personally have got that experience, and we got lifers where such cases have not given us oocyte, giving a trigger post 12 hours. We did the ICSI, the OPU again, retrieve 4 to 5 oocytes, M2 oocytes, good blastocyst, good outcome. So yes, it works. In such scenarios, you can troubleshoot. And with this, I'll move to the next question. Yeah, Manisha, please take it.
So, what options exist if sperm on the ICSI day is immotile?
I think, can I stop here? Can I interrupt here? I think just to continue what Dr. Manjunath has said, Nishad, I think times where the first 2 or 3 tubes are empty, I think first and foremost thing is we should also give a confirmation that cells are there or not, any cumulus cells are there or not. Just is a plain fluid. Then I think if there are no cumulus cells as such, then I think we need to troubleshoot the machine first. The pickup machine is right or not. The water home pickup, the suction pump is working or not. Then I think if there are no cells at all, that is a point. Otherwise, if there are cells and you don't get any oocyte in that, then I think it has been already discussed.
Yeah, you can actually check it from the quality of the granulosa cells and the whether there is hint of cumulus. If there is no hint of cumulus, nothing, then it is clean and clear. Also with HCG, when your trigger is HCG, these are the 2-3 things that you can mix and then decide whether to go in for the trigger and then post 12 hours.
Yeah, and moreover, like the timing usually never miss, and the administration will be also will not be missed. It's more of the potency of the drug. Sometimes it won't act properly. That's where the problem starts.
Absolutely.
If the cold chain is broken, if
something like that.
It's not administered properly.
If the patient takes it by themselves, most of the time that can be an issue also.
Correct.
They are not in our control. So yeah, so we need to take care of that. Yeah. So with that, can we move to the next one?
Yeah. So I was again repeating the question. What options exist is if a sperm on the XC day is immotile or has extremely low motility. You have seen it earlier. It was looking fine, okay, moderate motility, and now you are saying most of them are immotile or very poor progressive motility. What will you do with such samples? May I start with again, Akash? Are you there? Prateek, would you like to answer, or Rahul? Sanket?
Yeah, I can take it.
Yeah.
Pratik, go ahead. After you all.
Yeah. So in a lot of cases, especially if the motility is low and we already process the sample and then we put them in the incubator, the CO2 incubator usually helps with the hyperexcitation. So that's something that you can still get a little bit of it. And whenever we consider them for ICSI, motility as a whole point becomes a little moot. So in that case, even if you have sluggishly motile sperms, but you can track them, then I think you can just pick those sperms as long as they're viable. Because if you don't have any other option, that's what you have.
Yes.
And what you can also do is, if you're into using AI softwares, you can try SID and have the software pick the sperms for you.
That is something very controversial. Yeah, Dr. Jyoti, I would
see. I mean, sometimes because of the reasons like non-availability, some accidents, some performance pressure, we've always made it a rule to keep a backup sample.
Absolutely.
So, of course, pentoxifylline has to be available in the lab for the embryologist to look into it. But a backup sample, if it was fine, we always, as a rule, keep it. There is no option of not having it in our lab in today's times.
Absolutely. That is the best straight. But in case of a total immotile sample, if we have to select from that,
I completely agree with Dr. Jyoti. I think backup sample is the way to go. I think nowadays we are, and I think on the early first 5 days of simulation is going on well. Then having a backup sample is a must. So go for backup sample.
We don't begin unless, day 2 sample is not frozen. I take a written consent that I will not be responsible for the added cost which will come for on-site freezing in the lab. Why should we, get into it? We have a printed forma for that. If it is not frozen,
so let me just say this, that now consider that it's
continue. Just another two points to continue.
Sanket, just one moment.
Can I make a point here?
Yes, ma'am. Very well.
One small point.
Yeah.
I am not so much worried nowadays because we can always freeze the eggs. But of course,
but then the cost goes up.
But of course, we have to be ready. I agree with Jyoti. I always take a frozen.
I take a consent for that.
Yeah. So we should not worry too much, actually.
Yes. So I don't want to worry when I don't find
now
it
I
in
would
the
like
lab.
to take a different scenario. Sperms are there, but they are non-progressive. So what would you do? You will go back to the frozen or you will like to select from there? Yeah.
The point is, this is a very hypothetical question. Firstly,
part of the question that you missed is, it's a very bad SOAT sample. Frozen sample is not going to yield good results.
I was coming there. Nishad.
That is why I agree that everybody does that. So the question is, SOAT, severely compromised sperm, freezing of those sperm are not going to yield post-thawing good results. That is why the question is, what will you do in such case?
Yeah, can I continue?
Sure.
Yeah, still, in spite of that, you should have a backup sample. Number one.
That's fine. That's fine.
Yeah. Number two is you always have pentox or theo. I think any sperm motility enhancers, I would say, I think pentox is not cleared by FDA, so it's always preferably go with theophylline. Yeah. So that is one thing. And if you don't have a theophylline on your refrigerator, then please go for HOS. You can always do HOS. If there is a curling of the tail, then I think those are viable sperms in spite of not showing motility. Those can be used as a rescue. Last but not least, I think Rahul had a lecture recently in last microembed, laser-assisted sperm selection in case of immotile sperms. I think, you talk to the consultant and you talk to the patient. So we are left with only this option. There is no theophylline is not working out, and even HO is not working out. Then, last but not least, I think you can go with this with the proper counseling and consent from the couple.
For the freezing?
No. Laser-assisted sperm selection.
If
theophylline is not working, HOS is not working, and sperms are not reactive to any of these chemicals, then you can go for this as a last resort, but with the proper consent.
Yeah, 2-3 issues which arises when you start freezing for all the patient instead of selectively freezing in like patients who are from outside. Is there any difficulty in collecting the sample? Is selectively freezing versus freezing for all the things? One, it will be too much workload in a high volume units because that becomes a thing. The second problem arises is there is once you freeze the gametes, there is no clear cut in ART rules that when to discard. So that becomes a big headache. So we need to take care of that.
I have a counterpoint to add to that. I have a counterpoint. I think that should not be an excuse to not freeze the gametes because we can keep them for 10 long years according to the law. And when it comes to the workload, there are set guidelines on how much team members you should keep after a certain amount of cases so that the workflow is optimized. And if there are SOPs in place, it would flow very easily. And freezing semen samples isn't a labor-intensive work. It can be done rather quickly.
No.
So generally,
I'm not telling we should not freeze, but selectively freeze, not for every patient. That's
no, I think for sperm samples you should freeze it for every patient. Because
so
I think
I would like to hear from the experts because we have to
So my protocol is freeze for all and semen sample irrespective of the count and all that, because yesterday only I had a case. It was a 90 million count, but due to stress that man was not able to collect sample on demand. So that is one of the thing, because the wife is undergoing procedure. And another thing is when we freeze, we tell the patient that this is freezing going to be, because it is under our IVF package. So we don't charge extra for the freezing, and it is for 45 days. So we take discard consent at that time only. So 45 days irrespective of things, because by 45 days we'll get the result for the patient. If it is fresh transfer or something, if it is frozen, we'll ask patient that how long you want to freeze. And according to that, we give the reminder and we discard the sample. So that is simple.
Yeah, ma'am, that's the problem. ART rules doesn't have a separate consent for discarding those.
No,
You have to separately make a consent. Which may be accepted, may not be accepted. That's the issue.
No, as ART says that you should have the two kind of reminders to the patient, and these patients are not like onco patient or something. These are IVF patients.
That is the problem. So you need to remind 2 times for every patient. Then that is the problem.
Discard format, if we take it the signature, then there is not a problem. No.
It is actually while doing it only, while freezing only, she is taking the consent. That is correct. That is what we also do it at our center. That freezing of the semen sample, we do it at the time of starting of the protocol only, and then we continue it. And discard consent is taken after 2 months or once she is conceived. That's it. After 2 months mostly it is, and they give the consent immediately.
Absolutely.
And that semen sample, our container is also separate, so we don't have to search.
Right. So I think we should move lots of questions.
Nishad, I have one to make.
Yeah.
Ram.
So basically, if you go by the ART law, so there is a consent for sperm, gamete, and oocyte freezing. In that, they have clearly mentioned in the last section that for how long do you want to preserve your sperm, gamete, or oocyte. So in that, we can mention that if it is a temporary, maybe like as Dr. Charulata said, that we can write 45 days, we can write 60 days. Suppose that we can discard those samples. That is the number one. Number two, as most of us agree that we should. Sorry.
You can write
for a backup sample. Yes, of course, we can mention that into the consent form, which is
patient feel more confident with
it is
that
legally
consent,
accepted.
that their samples are not being misused, because sometimes they have this set in mind while you are freezing.
Right.
Exactly. And second point is like we have a lot of technologies now. I would like to highlight the sperm vitrification device. If we know that if the sample is cryptospermia or a low motile sperms or the sperms which are, I mean, like if we have only 5 to 10 sperm in whole field, we can pick those sperms and freeze into sperm vidi device and we can keep it as a backup. So we don't have to, freezing of oocyte can be as a last resort. So there are a number of techniques apart from pentoxifylline, HOS, laser-assisted hatching, and all this.
Right. Absolutely.
Sometimes now just one quick thing I would like to say. Sometimes practically I faced two things. Sample was good, but on the day of pickup, the sample was immotile. Then I realized that the patient had put his saliva in it. So all sperms died. We got another sample, and that sample turned out to be good because the volume was less. The patient thought the volume was less, so you can't get sperms. So there are
Exactly. The second collection is always there is.
Easiest solution is to tell the patient to give one more sample.
Yes, absolutely.
So that is one thing. You can start with that, and the rest that you all have mentioned have been absolutely wonderful points. And with that, we'll move to the next question. Yeah, so how do you handle oocytes with dense sticky cumulus that resist denudation? So what kind of oocyte it is? Can anybody tell me here? If it is the cumulus is very sticky.
It is usually a post-atretic or a post-mature oocytes where the cumulus is very sticky, very close, and there is no radiation or the corona radiata appearance that happens.
Yeah, so it's actually, it can be an M1, M2 also, the other way around.
Yeah,
because it's very sticky, very long, it has not developed that much. So obviously it's a sign that it's an immature oocyte also. Not always.
JVs and M1s.
Yes. Yes. So in such a case, how do you handle this situation?
See, Nishat, here, if it is like we always use, little bigger side 150 or 175 denuding pipette. But as long as if you are, polar body is visible, the little cumulus is okay. The other thought of school is that if you even do ICSI with the cumulus around, the developing embryo will be little better. So I do not bother if it is polar body is visible, then completely denude and blend up into the kind of degenerating the oocyte or something like that. So that is one point. Plus the repeated pipetting with the enzyme can help her, or what happens sometimes such kind of oocytes we can keep we generally this 80 IU highlase enzyme is there. You dilute so that you can keep little more time also. That is not a harmful to the, so that it can digest the cumulus, and then you can pick up probably the. So there is two things. One is leave little cumulus if polar body is visible, no botheration. Next, anyhow, when you do the kind of PN check, it will be stick into the bottom only. So I don't think it will be a bigger issue with this kind of oocytes.
Right. So rather than a rigorous denudation, we should take care of zona. We should prioritize saving zona in such cases rather than doing removal of the cumulus cells.
Correct.
Yeah. Also, OCC grading in general has been tried on by multiple people, and it's not very accurate because even the grading that came out, which was I think the most used, is a binary grade, 0 or 1. 1 if all the cumulus looks great. If it's not that, everything else gets put into 0.
Yeah. Great. So I think we'll move to the next question. We've got a lot of them. Yeah.
If high rate of oocyte degeneration is observed post-ICSI, what should be investigated? Oocyte degeneration post-ICSI. How
you're embryologist.
No, this is most heartbreaking.
Absolutely, that is the first one.
And after that, if the embryologist is same, the one who is doing ICSI with you since long, then what would you do? It's not all.
That's why I showed the first.
Why don't you think about egg quality? Sanket, Ramprakash, are you agree?
I have a counter question, Manisha. I think for how many cases will this apply, this question? Only for a single case, or say 5 or 10 back to back?
I think if you're seeing that back to back, you're getting decent amount of degradation, degeneration. So in such a case, obviously.
With a
I would like
validated,
to hear your answer in both the scenario. If you have
Yeah.
found it in one case, then how will you tackle it? And if you are getting it repeatedly, then it's something very, very alarming. You can't tolerate it for long. So what all SOPs you will follow?
So
so
if it is
basically,
happening only for one case, then we have to check the patient's gamete quality. But if it is happening for multiple cases, we have to check our procedure steps. Sometimes what happens, the ICSI needle what we are using, that sometimes if you change the vendor and if you are not properly aligned, that kind cytoplasm aspiration and it will come out. So we have to see that what is the size of inner diameter and outer diameter of your ICSI injecting needle. Plus the disc preparation, sometimes PVP or all that, if it is not properly maintained, it might, kind of change the environment for the thing. So that procedural steps plus instrument steps should be checked thoroughly, even if it is handled by the like skilled embryologists, but sometimes it may not be, ICSI dish may not be prepared by them. So this all things has to be, kind of checked that whether it is a procedure error, gamete error, operator error, or a media error. Sometimes, media, what they are loading is like not the HEPIS, but there's some IVF media. So it cannot be kept more outside. So all this, what very precisely every point has to be addressed.
Absolutely.
And anybody
Site
would like to,
quality. Yeah.
anyone would like to talk about anything about temperature of the stage or the media? Is it important?
I think temperature is the
before going to temperature, Nishad, I
the
think,
first culprit.
yeah, I
But
think,
also,
yeah,
there has
So
been
what
an
do you-
interesting case where
Yeah.
a certain disposable has been responsible for low success rates and post ICSI, and if you keep them in the incubator, because there was a certain company, I won't name them, but they used to manufacture their ICSI dishes somewhere else and the lid somewhere else. So now they had a problem in the lid with the manufacturing, and every time people would do ICSI, they would see degeneration, and there was low fertilization, there was just no growth. Even now, when that company sends their ICSI dishes, you will see on the back, it's mentioned very clearly now that it's not for use in ART procedures.
Yeah,
Yeah, anybody can take a guess which company it is.
definitely. So yes, you have to be very careful when it comes to the temperature of your stage, of your laminar mode, and even if the AC is on, many a times, the labs are small, everybody doesn't have an AHU. You're doing your ICSIs and the cold whiff of air is coming. You don't know if there is a temperature change. A small difference in temperature change can destroy all your oocytes, even if you're doing good ICSI. So you need to have SOPs and checks in place. That makes a lot of difference.
So before starting with embryologists, start with oocyte quality first and document everything. Here, documentation of the quality, the timing of ICSI, the media which was used in the cases, all such documentations are going to help largely. So coming
So
on.
alpha consensus says that less than 5%, if it is a benchmark with denuding after, so that also we have to mention and keep an eye. Yeah.
Right.
So that is a test even for the embryologist, how good that person is, that it should be less than 5%.
5%.
So regular check on your embryologist whether the degeneration rate is less than 5%, then you should allow that embryologist to do it.
Nisha, less than 5%, not in one case, in series of cases.
No, obviously not. I'm saying about obviously a cohort of cases, maybe at least 50 cases should be a good standard here where you can actually determine, no doubt about that.
and also one more point to be noted is, see, the moment you do ICSI, nowadays we have been masters of ICSI, so we take say 10, 15 or 20 oocytes at a dish. So once you have done ICSI, say for example, first set of the oocytes, say you have taken 10 in one drop. So I think those which are tend to degenerate will immediately start after say 1 or 2 minutes. They'll start. You can see very clear discoloration of the cytoplasm, right? So in that case, I think better you check your angle first. Your needle is sharp, or your injecting needle, is it blunt? Maybe sometimes people in a hurry or in high volume centers keep doing cases back to back. So it's always better. Sometimes there'll be fault from the company side. from the manufacturing end, so that that has to be checked. So just have a quick look on the oocytes that are already injected. If there is no degeneration, then please proceed. Otherwise, I think you have to hold back and have to check everything.
Perfect. So with this, we have all the things in place. Can we move to the next question, Nishad?
Yeah. So what should be done in cycles with no fertilization after IVF or ICSI? Both scenarios, in IVF and then in ICSI. Yeah, so I think Deepti can take this one.
Yeah.
I would like to know
Assuming
about
that
IVF.
oocyte and sperm are normal.
Yeah, so in IVF, what will you do if there is total fertilization failure after doing IVF? What might be the reason and what would you do in such a case?
In IVF, if the sperm count is normal and if the oocyte count is good, but still we can't
How
see
do you determine
any
the sperm is normal? Is only count a factor? Is sperm
based
count
on WHO criteria, if the count, motility, and morphology are seem to be normal, then we consider it as if they are normospermic samples, then only we'll go ahead with the IVF. In such cases, if there is no fertilization, even then we should go ahead with rescue ICSI probably after 12 to 14 hours. If you can't see any fertilization, then we should go ahead with rescue ICSI.
Okay. Anybody who would like to add something to this?
Yeah,
It
I
was
think first
already
and
an
foremost
ICSI
thing is,
cycle. We could freeze
I
the
think
oocyte again.
first and foremost thing is you should categorize the case here. Is it a first cycle that a patient is undergoing or not?
Yeah.
So if it is not, then please check back the history, get the report and see whether what is the fertilization rate of the previous case. So that is one thing. Well, if it is happening for the first time, then you check your all the parameters. If nothing is to be found wrong, then yeah, even the guidelines, I mean, the recommendations say 1 to 3% is noted, either it's ICSI or IVF.
Right. Dr. Jyoti would like to add something from the
I was saying that if
clinical
it was an
perspective that
IVF
really
cycle,
matters.
of course, as Deepti said, rescue ICSI could be planned. But if it was an ICSI cycle already done and yet you find no fertilization after 18 hours, then the question would be what next? In that case, just freeze and reevaluate your case and see what went wrong. Maybe the semen sample looked normal in all parameters, but DFI was not done. So look at the DFI and give antioxidants
Right.
for 6 weeks and then replan it.
Exactly.
And
So
one more point is, Ishad, one more point is, one more point is, if it is failed cycle, I mean, failed fertilization in the previous cycle, then yeah, there must be definitely
we are not talking
reason
about
reason
the
of
first cycle itself. So we are talking first cycle itself. Then we move
first
to
cycle is a different thing. Otherwise, there'll be absolutely PLC zeta factor coming in here. Then you can go for oocyte activation.
But
based on
does it
calcium
really help? What is your honest experience, since all the embryologists and clinicians are here? What is
Shall
your
I add one thing here?
Sure, ma'am.
Yeah, please
So
go ahead.
if it is a first cycle case and you have planned for complete 100% conventional IVF, which is a very, very rare scenario because 95% is all ICSI. In that case, rather than having a troubleshoot, why can't we do a sperm survival test, which is very easy to do, so that if there is a sperm is survived for 48 hours into the laboratory. So that is one of the criteria to check that the sperm has a fertilization capacity and it will be surviving 48 hours.
I think, Charu ma'am, see, survival is one thing. What we are trying to prove is the functional capacity of sperm to fertilize. So if even the count is maybe, let's say, 120 million, 150 million, and if the DNA is fragmented, the PLC zeta is not going to go out, it's not going to get activated. So how are you going to
Very true. But most of
tendency
the
to exceed?
cases in conventional IVF, when we were doing, almost all conventional IVF and only indicated cases ICSI, sperm DNA and PLC zeta plays a role. But most of the time, if you check the disease of the total fertilization failure or very poor fertilization failure, you can see the dish that sperms are not good survival rate, whatever it is the reason. So this is one of the thing I am telling. It's not that we cannot, in such case we can always do 50-50. Plus, other thing is rescue ICSI with short-term incubation. It is now, we can say that two polar body and 4 or 5 hours short in insemination. And now we are doing also HIC, which is high insemination concentration. And you change the media because short insemination, and you check
4
the
hours.
two polar body. That is also indication for the fertilization if you are not clearly visible polar body with NPBs.
If I can add something, I think whenever total fertilization failure happens, the first step is always to look at if it was one patient on that day or the whole cohort of patients on that day. Based on that, we'll take the steps. And if it's just one patient, if it's all patients, then a lot of things have to be taken care of, especially your lab, your embryology skill. A lot of things have to be now tracked. If it's just one patient, then, and if it's the first cycle or the second cycle, I think this is where the role of add-ons come into play. You have some tests which are subjective in nature, DNA fragmentation for, like an example, which you can do. What you can also do is, if you're worried about the DNA integrity, like Nishad already mentioned, you can also opt for a PICSI. It will not improve fertilization rates, but what it will do is will ensure that the sperm that you have picked, it has integrated DNA.
This is again a debatable topic. I would not discuss it here. PLC zeta, artificial oocyte activation, and PICSI. These are
The problem with people considering add-ons is that they think that it should improve fertilization rates, implantation rate, live birth rates for
fertilization. That's the problem. The problem in our country is that we go with fertilization rate. We don't go with implantation and pregnancy rates. That's the problem.
But
because
also
fertilization rate is 30%, but if you are getting 2 blasts, it makes more sense. Here, the first criteria in any clinic or with a clinician is, do you get 70 or 80% fertilization rate? Nobody talks about the blastocyst rate.
Exactly. Exactly. And also add-ons are supposed to help in specific, very specific, times where they can improve that situation for just one patient. It's not required that you do it for every patient, but even if it can help one patient, I think it's a worthy add-on.
You would like to do a PICSI or artificial oocyte activation in such previous failed fertilization cases.
Yeah, now there are a lot of treatment options that you can give, specifically if it's just one cycle that has failed for a patient and not the whole cohort. If it's the whole cohort, then of course you look at
Then the lab factors are there.
Also, one important factor is the age. Age of not only the two sides, but age of the man. There have been lots of publications. If it is obviously the cohort has been Caucasian males and Western paper publications. But if you consider what situation we have, it's around, if the male is more than 40 or 45, the chances of fertilization are going to drastically decrease. And in such scenarios, it is quintessential that you do the DNA fragmentation or treat the patient with at least 90 days of antioxidants, and then you do the cases. So thereby you reduce all these PICSIs and all the other add-ons that are necessary. Rather than doing a jugaad kind of troubleshooting, you can do a true troubleshooting.
Exactly.
By preemptively sorting this
So here you have to work on a detailed history rather than age. His lifestyle and like a lifestyle modification is probably you want to talk about, because excessive smoking and drinking can enhance such factors again, apart from age. Yeah.
Yes, very true. So moving to the next question. Yeah, Manisha.
How should abnormal fertilization outcomes like 1 PN or 3 PN be interpreted and managed? If 1 PNs are there or 3 PNs are there, how you are going to manage such cases? If they are there for few embryos, how you are going to handle them? What would be gonna your stand for such cases? How are you going to keep them separate, or how you are going to treat them, or totally out from the culture? You are not culturing them, or would you like to culture? How would you go ahead with such cases?
Can I come in, or anybody is answering?
Yeah, please go ahead.
So now whether we should see that what is the mode of fertilization, whether it was a conventional IVF or a ICSI. So now another case is 1 PN, 3 PN, and there is now another called 2.5 PN also. You know, some small PN. It is also there. Yeah. So there was a wonderful lecture few days back in one of the conference on that also. So now even in ICSI, if you are injecting 2 sperm, because now all skilled embryologists are taking 5-6 oocytes, and if you inject 2 sperm in a 1 oocyte by chance also, then also there is a chance that it will be a 3 PN even with the ICSI. And 1 PN is also, but such, if we have to see the number of oocytes available, if rare 1 PN is there definitely, or 3 PN is there, or 2.5 in this, my policy is that I keep that oocyte in a separate droplet and culture, so that many studies says that they have checked with the PGTA and they said it is euploidy status they have checked, and it converts into blastocyst rate also. But every patient cannot afford PGTA, and if there is the only source of embryo, then only consider to be transferred, otherwise no.
Right. Manisha, you're on mute.
So anybody else would like to add something to what ma'am is saying? Any other?
Ram, would you like to?
I think Ram is not there. Sanket?
Yeah. Just to extend what Charu ma'am has said.
Yeah.
I think, yeah.
Your voice is cracking,
The
Sanket.
point
We
that
can't
you
hear you.
noted, like, yeah. Actually, I'm traveling. Yeah. Sorry for that.
No.
Yeah. Is it okay now?
Yeah, absolutely.
Yeah. So the point is, yeah, absolutely agree with Charu ma'am. Only point is, in case of non-2PN zygotes, either it's a 1 or 3 or 2.1 as such new thing. So better to culture them separately and allow them to grow till blast, because then that's the way to go. Because at times what happens is, most of the time syngamy takes place in only very few quart of oocytes. It takes place very early. I mean, there's a lot of data on that.
Right.
So they say that they have already passed the PN stage and they've entered to zygote stage. If syngamy stage, if it is 1 PN, so it's 3 PN straight away. I mean, you have to discard it, because sometimes the body, or there'll be extra sperm not even done.
Okay, I think we don't have the connection. Okay, quick question. So in such a scenario where Sanket just explained that it is a 3 PN, which becomes a blastocyst, and now you just got one single blastocyst out of this, are you going to transfer this? Can I have the clinicians here? Will you transfer this embryo if your embryologist says that it is a blastocyst, but it was a 3 PN and you cannot do PGT on it? What will you do in such a scenario? Do we have Dr. Manjunath? Dr. Jyoti, are you there?
Dr. Akhi, would you like to intervene?
Yeah, please.
Yes. Nisha, did you ask something?
Yes. So your embryologist tells you that I've got only one blastocyst of the patient, and it was a 3 PN. You can't do PGT on that patient, okay,
Correct.
for aneuploid. Will you transfer this blastocyst?
See, in private practice, it is very difficult to, tell the patient that this is not going to be there. But some kind of information needs to be given that, see, in any pregnancy, there can be some abnormality, but we'll keep a close watch on it. But you can't say zero transfer. You cannot say, okay, fine, everything went fine. So somewhere the counseling has to be done very gently, that there could be any IVF cycle. I would not mention 3 PN or anything technical to the patient couple, but I will certainly take a caution here and take written consents and counsel. Every counseling like this, we document it.
Right. So
because every good patient becomes a potent litigator today. You do not know. Today they are very sweet to you. Doctors have full faith in you, complete faith in you, and tomorrow that patient will see you in the court in good faith only.
Absolutely.
Very true.
I really appreciate your honest approach and very transparent answer.
Yeah. So I will be very tactful in my counseling. At the same time, not be very safe on it.
I know.
Right. So the key message here is that counseling has to be very strong. You have to have a good rapport with your team, your embryologist, the clinician, everybody, and then make sure
because it will act like a double-edged sword. Tomorrow, if you transfer it, then
get everything in writing.
Don't do it. You have cheated me with the money. Simple
You
as
can
that.
tell, yeah, the patient has to be informed that if we transfer this embryo, the chances are that it can have some mutation or genetic defects will be there.
Many a
Get that in
times,
writing and only then transfer it.
many a
Yeah.
times they come back with some ultrasound ecogenic foci in the heart, something else there. You have to only keep reassuring them, and sometimes there's nothing. It's overrating in the ultrasound also these days. But a word of caution and a written consent in such cases is a must.
Right.
Absolutely.
All right. With that, we move to the next question. Right. So, what is the response protocol if a culture drop dries out or shows microbial contamination? What will you do if your dish, the media drop, has evaporated or become smaller? This is first scenario. Then we'll move to the contamination part. Yeah. So, is Ram is not there. Deepti can take this one.
Yeah.
So,
No, yeah, please, Ram, go ahead.
yeah, Deepti, go ahead. Deepti, go ahead.
Okay. If we see the droplet is dry down once we cultured, it is better to not to culture the embryos in those culture medium, and I would prefer to shift them into a fresh medium where the drop
Right,
is properly done.
but if the scene is that when you realize that it has dried out, you have to equilibrate the media for a longer time, then what will you do in such a scenario?
Actually, we normally have extra medium in a tube, so whenever it is a situation, so we can get it done immediately. So if we don't have any culture medium, then it will be a very difficult situation. Probably, if we are using something like 4-well, if one drop is dried out, you should try the other droplets.
Yeah, okay.
But
Fine.
still,
So
I
first
would
of all, why
try to transfer them as early as possible to a new dish. And if the
first of all,
contamination
Nishad, we will
thing,
not land up into. Yeah.
yeah, please continue.
May I come in? May I come in? So,
Yes,
Nishad, first of all, I would not let this situation happen because the standard operating procedures, SOP, is very, very important while we make our dishes. So, the person who makes the dish is the responsible for this scenario, and we should be very, very careful while they are preparing dishes. Either they are preparing on hot surfaces, cold surfaces. What is if they are preparing on hot surface? Are they using petri dishes beneath the dish where they are making, or I mean, it is one of the I would say drying out a drop is a crime done by an embryologist. The reason is why am I telling as a
Very
crime
well
is
said.
if you put embryos in this and it is at a stake because if they have not made it properly or if they have not overlaid oil properly. So these are the two possible reasons why the drop will evaporate or they completely land up into this situation.
Absolutely.
Right.
Very
Also,
And also, I
I
think
have a
second point is check your incubator culture. If the temperature is really high and you are not overlaid with enough oil, and also your droplet size is very small, then you're gone case. I think the droplet size, there is a whole set of theory. What should be the immunodensity in a particular drop? You're going for droplet culture.
Also, do you think that the water that is used in the incubator that makes a difference? That is used for humidification.
Modified incubator, it slows down your operation a bit slower when compared to dry incubator.
If it is properly overlaid by oil, then Sanket, I don't think so. If it has anything to do with the water inside the incubator, because even in dry incubators, we make drops and we culture them, right?
Correct.
No,
I
but
mean to
there is
say,
evaporation in dry incubator.
yeah,
The rate of evaporation is higher.
Slightly
also,
higher.
in that case, I have a point to make, which I think one of our moderators will be very happy to hear. Use heavy oil.
Right. So if you are using dry incubator, then heavy oil. That is
And one more thing, we are in a habit of extended culture. We don't change the media on day 3. It is quintessential to change the media on day 3, come what may. Even if you are using a time lapse, everybody uses it.
Single step culture, but change it on day 3. And don't use very small droplets. So coming to the contamination part, if you see contamination, what will be your mode of action? Prateek?
If we see the contamination, I think even after shifting and even after repeated washing, what I have seen is the contamination, the growth still appears.
Yes.
But repeated washing and continuing them till we can get the blastocyst, and we can culture the we have to culture them separately and we can freeze them. but repeated washing, and we have to shift them repeatedly so that otherwise the embryos will be degenerated. That's what I have seen in my personal experience. So but still we
Can
have.
I comment? Can I comment here?
I'm waiting to hear some other opinion as well because
Yeah, can I comment here?
yeah, sure.
Yeah. The point is, first we have to rule out whether if you're practicing a droplet culture and you've cultured only to say 2 to 3 oocytes in a drop, just make out which droplet is contaminated. Number one. Okay. It could be because at times it could be process mediated. While preparing a media, I think your dishes, I think the tip of the microwave might have touched some surface or your hand of some way if you're not wearing gloves or preparing the dishes. So that is one thing. And the point is, the moment you renew with fresh media, it's like throwing the microbes into a fresh food zone.
Fresh food, yeah.
They'll multiply in a rapid way. So the point is, the culture, what you get to see visually, will reflect only after 24 hours.
Right.
For example, you're washed today. For example, say if you're using 4-well dish, there's a contamination in 3rd well. Rest of the 3 wells are doing good. Don't disturb them. Leave it as such. No need to bother about it. The 3rd well, what you encounter in case if you're transferring them to a fresh culture media, I think that's a blessing for the microbes.
Okay, so you would like to go for only one droplet, and you will keep remain all remaining droplet continue with the same culture. You will, but there might be risk of contamination in those
So
droplets.
what you will do if all the droplets are contaminated?
Then though you have
See, that also depends on the degree of contamination. If it is too much, you can't do anything anyways.
The degree, I think it will increase by day. Probably on day 1 we have seen little bit. By day 2 it might have completely taken over the full drop.
so that they will see.
Anyone is sending that contaminated culture media for
Yes,
test?
that is exactly what I was mentioning.
Exactly.
If we send it for testing, see what microbe it is. That's
Because it is not only bacterial growth. I will tell you my experience. It is not only bacterial growth. Sometimes it will be yeast or fungi also. You know, I had a troubleshoot in one of the laboratories. So ear edge is laminar flow. They have never changed the filter. And that fungal growth used to come, that spores used to come from the top because they said, so that we have to check it. And I know that how I have done that. I have myself has made a rose Bengal agar dish and check that this is a fungal growth.
Right.
So one has to be careful that, turbidity and all that, mostly it is a bacterial contamination, but we have to rule out by sending the sample that what contamination, and then you have to thoroughly disinfect the lab.
Right.
Yeah, if you're seeing a lot of cases in which this is happening, then also please check the air quality in the lab, filters in the lab, because a lot of times even they are compromised.
Actually, even the ducts of your air purification system, they are the main culprit. They are never getting cleaned so thoroughly. You are changing filters, you are maintaining everything, but those ducts are the main source of spores and fungi infection.
If you are having an HVAC, then you need to make sure that the ducts are cleaned properly.
Oh, absolutely. It's a headache, but you have to do
That is
it.
the biggest source. People put in AHUs and they think that they're bringing out good air. Actually, it's the other way around. You are actually incorporating more bad air microbes into the lab. So that has to be maintained thoroughly.
Absolutely. And
The
if you
the problem with contamination is, even though I think gentamicin is a ingredient that we get to find in the culture media, which has a very wide spectrum of antimicrobial effect, but the microbes, the moment they keep doubling for every 20 minutes, say, the entire pH of your media is gone.
Yeah.
No matter if you're renewing the media.
It is obviously, it's a chain reaction that what the microbes, secretomes are coming out, excretomes are coming out, that is going to change the pH and everything else, no doubt. So we need to be careful, make sure the lab is functioning properly, check the air quality, check your dish. Somewhere instead of doing group culture, try to do drop culture so that not everything is contaminated together. So these are the small things that you need to take care of. And obviously send a sample for culture to find out what kind of contamination it is there. So you can actually identify the problem and take care of it as the time goes by. So with that, we move to the next question.
And also, Nisha, one more point to be mentioned. See, when people have when they renew media on the day 3, I think we have to be very careful with the denuding pipettes, with your strippers. It should not touch anywhere. Otherwise, again, it will end up in process-mediated contamination.
Obviously. So coming to the next horrible situation, how do you handle sudden incubator malfunction with no backup system? You have one incubator and
one incubator.
so how would you go ahead?
Vitrification, first thing. Keep your vitrification. Wow, you're freezing media down. Please inform the consultant and your team. Freeze the embryos or whatever it is, whichever stage they are, either zygote stage or marvelous stage or blastocyst stage, whatever it is. Then think about your machine.
Perfect. Anybody else would like to add anything to that? Any expert comment?
Yeah, I think in times like these, which we are, everybody should at least have 2 incubators. Even if you have a small incubator, if you only have one box incubator, say for example, if you're not a lab who's doing a lot of cases, you can still choose to get a single shell or an incubator with just 2 wells, a small incubator, just in case something happens, because you always have to be, you always need to have a backup. This is not a time where we can be without a backup in our situations. I mean, we are going as far as to freeze every semen sample, should have a backup for every step.
Right.
Very true.
So quickly moving on, because the whole point of this question was have 2 incubators, because there are centers, small centers, especially in 2-tier and 3-tier cities, where they are having only one incubator, even when the law says that you have to have 2. The point is, it is quintessential. You should have 2, because such scenarios will happen and then nothing can be done. So always have a backup, because that is the heart of your embryology lab.
One more point I'll add here.
Yes.
Yeah.
Keep a good relationship with your neighbors so that.
Very true.
But how to
transferring
transport, ma'am? Problem is, howsoever good
whatever there in the incubator, that can be frozen. But suppose whatever case is ongoing or that can be transferred. I'm not saying that embryo you can transfer in the test tube.
What if I had a scenario? I had a scenario where the incubator just gave up and we had to pick up the next morning.
Oh,
yeah.
Oh.
So can I intervene?
So I will share one
we
of
are already,
my
I think, running short of time.
incidents
We are
which
overshot
has-
actually.
Fine.
But would you quickly, wind it up? I mean, I'm enjoying it though.
Yes.
Okay.
So
I'll just move on quickly and okay, I want to go to this question. The biggest problem, I think we all have heard about what has happened in Monash in New York. Also, this was quite long ago in 2019 that IVF mix-up happened, and they gave birth to twins, which had none of the couple had their DNA. It was somebody else's embryo that was transferred. In the same manner, there was another case. I think you remember this one. The husband thought that the wife cheated on him, and they had another problem because they had an Asian baby. Then later on, it was found that the IVF lab goofed up, mix-up happened, and as a result, this happened. And this has happened in many labs. I can go on and on. I'm not going into details of it. And ergo, how should you respond to witnessing errors? How important it is? And there are 2 scenarios. When it is gone, then something like Monash will happen. But if you have seen that this person's semen sample has been mixed, or even for that matter, when you're making multiple samples and your pipette is going from one semen sample into another, in such a scenario, how do you stop it? Where do you stop it and what is required? Charuman, can you?
Yeah, so nowadays, see, I agree that everyone cannot afford the electronic witness system, and most of the lab are not having. But India, we are a wide country and we always have a good, human power around us. So even if the nurse can be the witness, and if by chance, at the most for your SOP, I always say that do not even try to prepare more than 2 samples at a time. So collect the sample in a gap. So prevention is better than, kind of troubleshoot. So give a gap if you have 5 pickups at a time, but give a gap in the collection so that 2 samples at a time can be processed. and every pipette and all that has to be, we
Labeled.
have a marker, so it can be labeled. So that is, and if by chance, even if it is 1 in 1,000 times the error happened, immediately inform your senior so that it can be prevented at that stage only, and the second sample can be collected, or if we have a backup sample, that can be collected. And as long as embryo and all that things, never mix up that this patient has only two sides and we are using 4 wells, so we are just never one patient, one dish.
Yes.
Very true.
Absolutely.
Agree.
Absolutely.
Right. Anyone would like to add to this?
I would like to know the clinician's perspective. Okay, you came to know that some mishap has been happened in the lab, and we can't continue with such embryo, like we can't go ahead with ICSI or something. So how would you counsel the patient? Are you going to disclose this thing to the patient, or you are going to give simple cover up? Any clinician?
Rahul, would you like to say something? I think do we have Dr. Manjunath and Dr. Jyoti?
Dr. Manjunath and Dr.
Jyoti.
Dr. Jyoti, ma'am.
Okay, fine. So yeah, so this is basically what it is, that what troubleshooting you have to be, it's very simple. You can avoid errors. The most important thing, as Indian labs go, have a witnessing person, have 2 embryologists in the lab, record everything, take pictures of everything. Documentation is the key. It is your first and the last line of defense. So it is very important. Do single drop culture so you can actually trace which embryo is in which drop, mark everything properly, and obviously make sure that your procedures, the number that you're doing, are manageable. Not that you are 2 embryologists and doing 10 cases a day, even in a batch, because that will screw up the whole cycle. Mix-up can happen. Lots of problems can be created. Just one case can destroy the whole center and your career. So it's important. Have more people, have more incubators, record properly, document everything, and have a proper SOP.
Right. Absolutely.
Yeah.
So with this, we would like to.
So we'll just close it with this because we still have a lot of questions, but I think it would be
Time
nice.
is limited.
Yes.
So would you
basically,
yeah. Go ahead.
Yeah. So basically, the most important thing is the culture of truth telling, the communication, that you talk to your physicians. The physicians need to talk to the embryologist. If there are errors, own up the errors so that later on, for short-term gains, long-term losses, it doesn't work that way. Always make sure everything's recorded, documented. You speak to your seniors, juniors need to talk to their embryologists need to talk to the clinicians. This will foster a good culture, honesty, and this will actually end up in clarity, good working culture, and good results.
Here,
So
I would like to add one more thing, Nishad. Cultivate a culture of giving up, like confessing the mistake. A mistake can happen by anyone, and it's senior's responsibility to understand the scenario and protect a junior and develop a culture where they can come and tell you the thing. So you have to develop a very congenial culture inside the lab or between the team members.
Right, very true. So in conclusion, ART labs involve huge catalog of potential risks, and a cascade of catastrophes can only be prevented by acknowledging the risk involved in various aspects of functioning and management. So if you think that my lab is the best lab, it gives the best results, I have no problem. I'm sorry to say, don't be in a delusion. Be a realist. Optimism is good, but being a realist in IVF lab will pay you much better. So this can happen to any one of us. Be careful. Make sure all troubleshootings all done properly, all SOPs are there, have a good culture in the lab and the clinic. I think that is the only way we can move ahead and get good results. And with that, I would like to thank my experts and the panelists. You've been great. Thank you for this interacting session, and a good evening from my side and Dr. Manisha's side.
Have a good
Thank you very
evening, and
much.
I would like to thank Dr. Anuradha and Dr. Seema Pandey and the ESR series of webinars. It is a wonderful discussion today, and thank you so much, and have a good evening, everyone. Thank you.
Thank you so much.
Before anyone speaks, can I speak a word about this panel?
Yes, please.
Please stop sharing. Yes, Nishat and Manisha, what a mind-blowing panel. I was just glued to the screen. And guys, I accept that this is a very broad topic, cannot be taken care in one part. So we would have made it in two parts. So for audience benefits, I would like to have part 2 whenever we are going for our next embryology session with same set of moderators with different panelists, and we are going to complete the questions which you have left today. This was really, really very practical and something which all the already should be attending.
Thank you for
excellent. And Prateek and Charu and all the panelists, you guys were amazing. Thank you so much. You gave us a very, very good
Thank
show.
you for initiating such a practical discussion, ma'am. We are really, we had a good time being here with you all. Looking forward for the next.
Actually, I was having a good time. All the embryology was looking like a coffee table meet. You know, you are sitting and sipping coffee and you are discussing this. I loved it. And thank you, everyone. Thank you, Dr. Seema, for
I was in
providing
another webinar. I did my bit and I came back listening to all these young, bright and brilliant minds.
Thank you.
And thank you to our chief guest, Dr. Charudat, our guest of honor, Dr. Srinivas, and all our esteemed faculty. A big thank you to our partners, Corona Remedies, for this wonderful Core Connect program, which we are doing inside series and evening with the Sarians. And I would like to thank everyone and have a wonderful weekend. And also, I would like to invite you again, as Dr. Seema said, that we should, this team is fantastic and all of you are my favorites. So thank you so much and adios.
Your voice is gone.
Yeah, in India, we don't say goodbye. We say we'll meet again.
Yeah. Bye-bye. Bye, ma'am. Take care.
Bye. Thank you, everyone. Thank you, Nisha. Thank you, Manisha.
Thank you,
Thank you,
ma'am.
Dr. Anuradha. Thank you, Dr. Seema.
Thank you.
And
Thank you, Dr.
whomsoever I
Charu.
forgot to mention, thank you so much.
Thank you. It was wonderful evening. Take care.
And thank
Bye-bye.
you, Anu.
Happy
And
weekend.
Manisha, you were in another program where your name was there also. I was thinking, what are we doing? Just jumping from one webinar to another. So we are going to change the day of our webinar.
Please, thank you. Thank you so much, ma'am. I was hopping around actually.
I think, Dr. Seema, our next webinar should be Thursday because I am already in another 2 webinars. If you see my laptop side to side here. So let's do it on Thursdays from next time because Tuesdays, Fridays, we have enough programs.
Can you just go off air and please, participants, you stay. I just have to ask one more question.
Can we go offline, please? Offline. Mehul, are you there?
Thank you. We